Than here as I was running around this morning through the campus and through Corvallis on the icy roads. I mean, this is we can have this in the summertime. Uh, yeah, very short season. Um, a lot of zone 4, zone 5. There are a lot of fruit crops that we cannot grow in Montana, but we do have a great potential for specialty crops. We're only a state of about a million people, but 6 to 7 million of you guys come and visit us from around the world every summer to see the national parks. A lot of great potential for small farm, high value crops. We're also uh, another big part of our mission is there are some excellent growing areas in Montana. Great irrigation infrastructure. We're right there with the mountains, the giant snowpacks. So a lot of opportunities to grow those specialty crops, and especially with so many people moving into Montana. For example, around Flathead Lake in the northwest corner of the state, just last year, about 100,000 people moved into that area. Um, we've seen explosive growth, and that's putting a lot of pressure on our ag lands. And it's really through specialty crops that we can preserve a lot of these great agricultural resources that we have in the state. And it's getting warmer. Maybe someday we will be able to grow some. So I came over to the Western Ag Research Center or Horticultural Research Center in 2014 from the main campus in Bozeman. Um, there was no current faculty there at the time. They were thinking about shutting it down. We've really revived that place. We've got a lot of grower support. Recently had the berry growers in the state organized into an association. We've got a growing vineyard and winery association, a lot of new cider orchards that we all work to support. Anyways, when I came over in 2014, we started this project that was in general looking at uh, ways that Montana farmers could tap into the explosive growth of berries and blueberries, especially. There's a lot of people who are interested in high antioxidant fruits, superfoods. And so we planted out uh, about 50 different varieties in 7 different fruit species, uh, including aronia, black currants, red currants, elderberry. I'm forgetting serviceberry, edible serviceberry. Looking at which one of these had market potential for Montana. We're looking for things that are, zone 3 hardy, zone 4 hardy, uh, have that high antioxidant, dark color profile, but can be grown in neutral to alkaline soils because most of the soils in Montana are that way. We've got a lot of calcium in the soils. It's hard to acidify. Um, and yeah, we've learned a lot of great things about that. Um, they are all really cold hardy. We've had some record colds, including last fall in October when we broke a 100-year record for the coldest temperature in October in Western Montana. It went from it really hadn't froze all fall until late October, and then it was suddenly 10 below. Um, we lost all the fruiting buds in our vineyard. Many of our apple trees died back to the ground. But all of these plants did great. And especially the Hascaps we're really excited about, or honeyberries, because they are a new crop with a lot of potential. They have antioxidant potentials that are 3 times blueberries. They don't have any uh, insect pests or fungal disease. Um, it's really just managing weeds and birds, which is great. Uh, and in particular, we like the honeyberries or Hascaps because they have a great fresh market potential. They're really um, have a great flavor and also for processing. Wineries love them. They're great for jams and jellies. But out of all the cold hardy berries we're looking at, we're very excited about Hascap because it does have that fresh market potential. Um, so we've been growing we have one of the longer term and most diverse variety evaluations for Hascap honeyberries at our center. There are about 16 in the main trial, and we've got another 5 or 6 varieties that have been planted later. We've been looking at them for about 8 years now. So we're uh, getting that basic information that growers need to know. What's most productive? We're working with food scientists and processors to figure out which ones taste better, which ones the consumer likes better in different products from both fresh, and we've done single varietal honeyberry wines and put them out to different breweries and wineries and had them do evaluations to see if it matters. We've had great, we've learned a lot. I've mentioned a little bit about the hardiness. They're super hardy. Um, but we've also seen excellent production compared to other research. A lot of the other university research trials from both North America and in Europe are often showing yields in Hascap honeyberry of 2 or 3 pounds per plant, which ends up, and it's about 1,000 plants an acre in typical plantings. We've had much higher yields. We do take care of our plants. We've had yields of 4 to 7 tons an acre, which is certainly commercially viable. To back up a little bit, because I forgot to introduce the actual plant, who is familiar with honeyberry or Hascap? I know you two are. Everyone else, has anyone tasted him? You've tasted it? It's got a taste that's sort of a cross between a blueberry and a raspberry. Really, it's a great fresh berry. It is an edible honeysuckle. Lonicera cerulea is the species. It's native to boreal climates around the world. So from Northern Europe, Siberia, Japan, a lot of actually in Siberia and Japan, these have been cultivated for centuries. Also native to Canada, Oregon, Washington, Idaho, and Montana. And it's a relatively new crop. It was brought over, it was late 90s, really, when the first ones came over. Yeah, early 90s. And a lot of those first varieties were more, it hadn't really been, they were just taken from older varieties in Russia and Eastern Europe. Some of those initial releases really aren't representative of the crop. There have been a lot through the work of Maxine Thompson here in Oregon and Bob Boers up in the University of Saskatchewan. They really pushed this forward, combined a lot of the genetics from around the world to produce a much tastier, easy to manage, and more productive varieties. And I guess getting to the larger picture too, that the genetic resources for improvement in this berry are vast, and it's a unique berry in that it opens up areas like Montana that are cold, like too cold, don't have the acidic soils for blueberry production, too cold for blackberries, and it's just a great healthy fruit. So for the work that our center did for this project, we were really trying to wrap up some of the other loose ends in terms of the potential for honeyberry for fresh and processing markets. We wanted to look at storage life, because really no one had any idea. And so for raspberries, Randy, what do you expect for storage life? How quickly do you have to get your...
Well, the varieties that we use in the machine harvesting system have essentially 0.
Yeah, they're going right into the freezer. Yeah.
They're bred for maximum flavor and sugar.
Mm-hmm. But for a fresh market raspberry,
7 to 8 days.
yeah, fairly, yeah, and it's fairly short for a lot of berries. So really no one had looked at this for honeyberry or Hascap. So we did just some simple trials looking at both causes of spoilage, the length of time they can keep, which varieties keep longer, and how that would, yeah, and how long you can keep them. And I'm remembering that with my 3 slides, I do have some animations. So to review, they are really, and we also looked at machine harvest because there is, like I said, a huge variety in the forms and size of the fruit, flavors, when they produce. We are often picking honeyberries from the end of June to the middle of August because the bloom and maturation times are so widely dispersed. So in this is just a simple this small sample actually of the things we grow. You can see there are some like Wild Treasure and Sugar Mountain Blue, which are smaller berries, more Russian influenced. Kauai, Solo, Tana, Taka, Kiko, Chito. These are all Maxine's varieties, which are mostly Japanese. They tend to be rounder and a little bit smaller. And then Boreal Blizzard, Borealis Aurora, up there at the top, more oblong, larger berries, up to 2 grams of berry, so a really big berry. Those are varieties that come from Bob Boers' program up in the University of Saskatchewan. And yeah, so just a sample of the great variety. And again, we and a lot of the growers we work with are most excited about these because they do have that great, it's healthy candy. People love them. And yeah, we did this project in conjunction with our larger program to look at the commercial potential of these, to look at shelf life and potential for mechanical harvest. So for shelf life, the first thing we did was, nothing's known really about the ripening physiology. So what we did was, Rachel had done a little bit of work, saw that there was some ethylene produced by the fruit. Maybe it'll ripen a little bit more in storage. So we picked these at a half blue. So just when they're starting to turn color. And then we'd also pick them later when they're starting to soften. We see that sugar content stabilize in the mid-teens. We wanted to know, does it ripen? Let me see if this works. And those unripe fruit were about 40% smaller, and they do not ripen in storage. So they do get a little sweeter and the acid does go down, but they really did not change in a 2-week period. They cannot ripen in storage. But what we did see is, and I think this is really important for varieties like Tana and other Maxine Thompson varieties that are, when you pick them off the bush, they're a little bit sweet tart. And what we saw was that by leaving them for 2 weeks in a cooler, high humidity cooler, that acid went down and you're getting about a 1-to-1 TA to Brix ratio. So it actually tastes better the longer you leave it in storage, for a lot of those more sweet tart varieties. So how long can we keep them in storage? Well, these are kind of an interesting berry in that they're actually twin berries, like a lot of honeysuckles. And it's part of the reason that they have so many high health benefits. They have really 3 layers of skin because there's 2 berries inside an envelope that grows up around the base of the fruit. So there's a ton of layers there, a lot of phenolic compounds, creating great flavor, health benefits, and also, we think, affecting its storage life. Because when we stored it for in the cooler, so here's 4 different varieties over 4 weeks, really over a 2-week period, we had, and this is the incidence of fungal disease, botrytis, things like that, really had none over that 2-week period. And this is many, many, many clamshells sitting in the cooler. And we did this over 2 years and really saw almost no spoilage in 2 weeks. And some of these, we got out to 4 weeks with no incidence of any sort of fungal disease. really quite amazing for a berry. What we and really what will spoil a flat of these is shriveling and shrinkage, just dehydration. And this is overly complicated, but most of there is a big difference between varieties in terms of how long they retain that moisture before they start to kind of turn into little raisins. They're not getting moldy or anything, but they just start to look a little bit ugly. And this is the marketable tray life, which is the main thing here. That's when you had less than 10% of the berries starting to just show a little bit of sign of that shrivel. Aurora, which is a great tasting one, that could get you almost to 3 weeks. And the other ones were at least 10 days to 2 weeks. So nice shelf life, very little fungal disease, excellent storability of these berries. And this, again, is just for hand harvested, gently harvested fruit. Really for, and Rachel's done some great work looking at the numbers for labor in terms of what your return might be on something like that with that hand harvest. They are, we've also learned to wear gloves because this bloom on the outside will show all your fingerprints in a hand harvest. But they do keep well if harvested gently. So the next part of the project that the center funded was looking at not handling them gently. We want to abuse them like a mechanical harvester would. Are there differences between the varieties? And we do not have a $100,000 mechanical harvester. What we have is our own DIY harvest methods, which work great for a lot of our growers who are at the couple acre scale. It is a reciprotool attachment to a Sawzall, and we make these forks with our welder, and we just shake usually these bushes have 4 to 6 main trunks. In 30 seconds, you can go through and shake the whole bush into these trays, pour it into the totes or a swimming pool. Because this contraption costs $2,500, and this, if you go at the end of the summer when they're on sale, you can get it for $5. So we looked at this process, damaging the fruit in the field, and also the cleaning process, because you get a few bugs and leaves when you're shaking them off. And this is a leaf blower and a little slide we built. We were just looking at where the damage occurs. So we have this with 3 different varieties: Indigo Gem, which is an earlier rounder one; Aurora, which is the nice tasting oblong large berry; and Tana, one of Maxine's medium-sized round berry later in the season. We looked at hand harvest and the bruising and cracking rates, the shake and catch, which is just shaking it into the trays and then assess it there, and then shake and catch with the blower. And really what we saw is, yes, there are big differences between varieties going through the whole process, but also most of our damage is occurring in this stage. But again, this is just bruising and cracking. For processing market, wouldn't matter so much. We get it into the cooler, into the freezer, and it's fine. And we also did another simulated mechanical harvesting where we're dropping them from a great height and rolling them through some tubes. That also showed basically the obvious that smaller, rounder berries tend to be damaged less than that. But there's a lot more to be done in this. And like I began with, there's such a great improvement potential with the diversity in genetics and different genetic resources that could improve this fruit. And really, it's just been Maxine Thompson here in Oregon and Bob Boers in Saskatchewan that have started that journey to making these a more commercial fruit. And so we think the potential is great. And with that, I'd take any questions. Yeah.
Were these on drip irrigation?
Yes.
Okay.
Yeah, it does not rain much.
This is in Corvallis, Montana? Montana?
Yeah,
Okay.
it's in Corvallis, Montana.
Okay. Yes, thank you. Thank you. My name is Gabriella Tait. I am a postdoc in Professor Von Walton Lab here at the Oregon State University. And today I'll try to summarize briefly the results we got over the last few years, or actually the last summer of work. So here are our objectives. We wanted first to test our new deterrent in a semi-open feed condition and then try to identify by GC-adjacent volatiles emitted by the deterrent. Just to let you know what are we talking here about. We focus most of our research on Drosophila suzuki. It's an invasive species coming from the Southeast Asia that has invaded United States and Europe in the last 10, 12 years. So because currently our growers base their treatment intercrop on pesticide, our goal is to try to find new approaches in order to deal with this species, new approaches that do not require the use of toxic insecticide. We have been working on a new deterrent since 2019, and actually we are really happy to share with you these results because it's extremely excited the kind of data we were able to originate. Just to give you an overview about a deterrent is different from a repellent. So a repellent is something that you use if you really want to keep away an insect from a substrate, right? For example, when you want to keep away mosquitoes from your skin, you just spray repellents on your body, and this way you totally avoid the contact, at least for for a certain period with the insect. When you work with deterrent, you cannot avoid the contact between the insect and the substrate, but at least you're able to reduce the time spent on the berry by the insect. So actually, you still have an infestation, egg infestation on your berries. But if you check, if you have your control, you can verify that the mean number of eggs generally should be lower on your berries than in the treated berries than in the control. So this is what we are talking today. So Drosophila suzuki, invasive species, it's affect small berries like blueberries, strawberries, but also cherries as well, some varieties of grapes. And yes, and this is our deterrent is a mixture of few ingredients. All these ingredients are, most of these ingredients are plant-based materials plus some minerals. To get the formulation we currently have, we worked over the last 3 years over more than 50 compound tested, more than 80 different lab tests. Each ingredient has been tested for at least 3 different concentration, not only in the lab but also out in the field, at least in a semi-field, semi-open field conditions. This deterrent is the results of, as we were saying, 3 years of work done by myself and my professor, Von Walton. And because our involvement, we decided that it was extremely important to have a student and external people working and testing the deterrent in order to make sure that results were as much as um, you know clear and well defined. So as we were saying, the goal of this grant, one of the goal was to test the deterrent out in the field. We're really lucky to have some acreage of blueberries at the Lewis Brown farm here close to Corvallis. So thanks to the help of our student Ben and Edwin, we were able to perform big cages trial in order to verify the efficacy and longevity of our deterrent. So as you can see here in this picture, we were able to build different cages. This is just a schematic picture of what's been performed out in the field. And under each cage, we had the 5 bushes of blueberries. First experiment ran during August 2021, this last summer. We wanted first to spray to treat just one bush in order to verify if the volatiles emitted by the deterrent were able not just to protect the treated bush, but also to protect the other 4 bushes. Every week, we released 100 flies, 50 females and 50 males. We perform 2 monitorings per week, 2 sampling per week, and a total of 250 berry per time per cage were collected. And during September, we performed a second experiment, always using our deterrent. But if during August we tested just one formulation of the deterrent at 0.075% volume to volume, during the second trial we wanted to test 3 other different formulation. Why did we decide to go ahead in this way? Because if in August we tested the efficacy of the deterrent as just as was it, as a powder mixed with water, during for our second experiment, here we wanted to mix together our deterrent with with an adjuvant in order to get a better coverage of the berries. And for this reason, we tested 3 different formulation of the deterrent by adding the adjuvant. So let's go briefly through the results. As you can see here at the top in this first picture, we got overall real interesting reduction of eggs. If you here we have the mean number of eggs per time. So actually during 2 days we got an higher number of eggs on the treatment plants instead of the control, and we still have honestly to realize why this happened. So you can see a little bit of variability in between days, but overall we can say that our deterrent were able to protect berries from Drosophila suzuki. In September, we got still good results. If at the first day of monitoring, we got higher number of eggs on the treated berries. On September 13 and September 17, we were able to reduce the impact of Drosophila suzuki on the treated berries. To the best of my understanding, what happens here is then when you spray our deterrent, you need a few days before the volatiles and all the other ingredients can create a sort of barrier on the berries. So at least this is what my understanding, but for sure we need to understand better, when to spray the deterrent in order to have the best level of protection. And the second objective of our grant was to detect key volatiles emitted by the deterrent in order to identify and later think to create a second version of the deterrent based only on key volatiles. So we performed GCD analysis. What does it mean? GC stands for gas chromatography and ERG electron tenography analysis. So at the same time, we subjected the antenna of females and different antenna of females and males to a flow of air flow containing the volatiles extracted from the deterrent in order to see towards which volatile we have a physiological reaction of the antenna. As you can see here in this picture, we have some responses from both males and females, and our next step will be to do MS GC analysis, which stands for mass spectrometry analysis, because now that we know towards which volatile we have a physiological response, we need to identify and characterize each peak in order to proceed with our research. We got actually results also from some collaborators from New Jersey who wanted to test our deterrent this summer. As you can see here, OSU stands for our deterrent, worked really well in a comparable way to others' product. Actually, if Jet Egg is a commercial available product, which more or less the same aim of our deterrent. So it was extremely exciting to receive data from a third party, independent parties, showing us how our deterrent perform well out in the field. For sure, more analysis are necessary, and involvement of more people is required for a better understanding of how does work our deterrent. So to summarize, we know that our role is to provide growers new approaches, new technologies to deal with Drosophila suzuki. Just an example, we for an organic blueberry production, our growers currently have few available products like Venerator, Grandivore, Jet Egg, and Trust, but if you consider that they need to spray up to 24 times per season pesticide, that is help us to understand how is important to find new biopesticide, we can say, in order to provide more diversification between pesticide. So, which are the benefit for our industry? For sure, by having this deterrent out in the feed, we can have different multiple good aspect. For example, during the last 3 years of testing, we didn't reported any phytotoxicity on plants. We didn't see any negative impact on berries. We have a comparable efficacy of this technology to the common available pesticide. We are talking about 3, 5, 6 days of protection. So our idea is for sure to work more in order to have a longer efficacy of the deterrent. We would have a similar application cost, but still we have quite a lot of work to do. We need to make sure that the taste of berries, of treated berries, is the same. We need to involve more people, create like some taste label in order to involve people and make sure that the perception of the berries and the taste is the same. We want to, we need, as we were saying, to identify the key volatiles coming out from the deterrent. We need to do open field trials, and luckily we have growers here in Oregon, as well as California and other states, willing to go ahead and test the deterrent in an open field condition for this coming season 2022. So we still have a lot of work to do, but our data are extremely encouraging, at least in our mind. And yeah, this is it. If you have any question, just go ahead, and thank you for giving us the opportunity and giving us the money. So yes, thank you. Oh yeah, we have a question here.
I might have missed, how many days after you applied the deterrent,
Uh-huh.
how many days was your first sampling?
The first sampling were oh yeah. The question is about how many days after we got the first sampling. Yeah, thank you. Yes. So after the application of the deterrent, we generally wait for 3 days before proceed with the first monitoring. So between 3 and 7 days. Oh, here we have a question. Is the product available for use in commercial production? This is our plan. We need time. We are scientists. We need more time to make sure that the product, that the technology, as is it, can perform well out in the field. But our goal is to have this technology available in the future for our growers here. This is our goal, definitely.
So you have a figure there with 5 bushes. If you can kind of just explain to us a little better
Yeah.
how that trial was done, because only one of the 5 bushes were sprayed, and then how that impacts surrounding bushes as well.
Yeah, for sure. Actually, I don't know how to go on that slide. Yeah, sorry, it's just blocked. Sorry. Thank you, John.
That was kind of an interesting setup where we were just
Thank you.
there.
Thank you. Yes, here. Yeah, the question is if we can describe better how we did perform this experiment. So yes, we treated just one bush out of 5, and actually what we saw is that there is an efficacy, a protection efficacy also on the bushes not treated with the deterrent. So yes, so at bush, the closest bush, as well as the third and fourth and fifth, we got a similar number of eggs in comparison with the treated bush. Of course, the number of eggs was a little bit higher on the farthest bush, but still we were able to get a coverage also on the other bushes, not treated bushes. So this was, yeah, please.
So you sampled, at each sampling, you sampled 250 berries per the big
Per
cage.
cage, yeah.
50 berries per
Per,
bush.
per bush, exactly. From the bottom, 25 from the bottom and 25 from the highest, yes, top part of the bush, for a total of 250 per cage.
That work was not done exactly the same way in Rutgers. So the Rutgers data, that I think 2 slides further,
Uh-huh.
show the Rutgers data. That
Mhm.
one. That was done slightly different.
Yeah, the setup of this experiment was different. Here we are talking about smaller cages, one plant per cage, and berries were placed in small bags and hang on the plant. If I remember well, 50 flies per cage were released. So it was pretty different. And also here, if our method is always to check on the number of eggs, our colleagues there prefer to check the number of larvae and later the number of adults emerge from the berries. So, but still, this number shows us that on treated berries, we have a lower number of eggs laid. So,
I'm
yes.
wondering do you think that the fact that there's fewer bushes that are treated
Mm-hmm.
in the cage relative to the ones that are not treated,
Mm-hmm.
and you're taking the same number of fruits,
Uh-huh.
that there's so many fewer fruits that have been sprayed that it somehow influences the eggs that you get. You know what I mean? Like if you had like 3 and 3 that were sprayed and not sprayed,
Uh-huh.
there's like on average, there's the same number of sprayed fruits that
Sure.
are not sprayed versus
Sure.
your setup where you have 4 times more
Yeah.
not sprayed versus sprayed.
Yeah.
I'm wondering if
Yeah.
like
Yeah.
influence somehow.
Yeah, here we are talking about how the deterrent can actually work, at least in this trial, differently by spraying a lot of berries, bushes, big bushes, as in our trial, or just few berries, as in this experiment developed by our colleague. I guess so. I guess that for sure, when you work on different quantity of berries, results can be different. And
Because you take 50 berries from the sprayed bush every time, so there's less berries on that bush that are
Yeah.
sprayed, but you have 4 times as many unsprayed berries. So I don't know how that might, I don't know which direction it would go.
Yeah. I guess that we need to run some statistical analysis by including these perspectives. I guess that we can create a sort of model telling us, what does happens when you have big bushes treated, and step by step you just get a few berries here and there, or by doing an experiment like this. So I guess that is something important to find out. And yeah, it's a good question. Thank you. For sure, I guess that is something we need to talk with our colleagues, also in the future, to make sure to have the same kind of setup in order then to can compare our
Yeah, it's just
analysis.
a little unbalanced. Then.
Yeah, for
So
sure.
we have the choice and the no choice, and this is a no choice experiment here.
Mm-hmm.
The previous one is a choice experiment.
Yeah,
So you
exactly.
do that for different reasons. This one is kind of the tougher one of the two. The other one is kind of to see how little you can get by with.
Mm-hmm.
What's kind of interesting about this trial is the 2PF that you show there. That's very different than the OSU one. With the OSU, you have full cover. The 2PF, which what we were hoping for and hoping to improve on that, and that's not ours, it's a patented product out of Cornell. That is only, it's a sachet that's put in the bed on the bush. And so it's a repellent, right?
Yeah,
so
yeah.
The advantage of that is huge in terms of EPA approval and that type of thing.
Yeah,
That's why we're
exactly.
so interested in comparing these side by side, but all of the others, they have different modes of action. I don't know if you want to talk about that.
Yeah. Uh, uh, I guess that.
I think they're wanting to err with the side of questions and Q&A, so I think we're good.
Just 4 minutes, I guess.
Yeah, so here we are talking about how it would be easier to apply just a dispenser with key volatiles, so create a sort of a repellent instead of having a full coverage product as we have in terms of EPA registration. For this reason, we are trying to identify key volatiles emitted by our deterrent, because the next step will be to work to put together these volatiles, create a sort of a dispenser to place out in the field. So in this way, we will not have the necessity to spray the berries, but have the same efficacy level by placing like a sort of dispenser per bush, or every maybe 5-10 bush, and have the same efficacy. So we're working on. I guess that it takes a little bit of time, but we are on a good track here. And yeah, this will be a good goal also, because we really need to try to reduce the pesticide applied on berries, even if here we are talking about a biopesticide, so we don't have any toxic inside. Still, you have to apply something on berries, and this could impact on the fruit quality for sure, and also on our workers out in the field. So there are many many parameters we need to consider, and for sure, thank you for pointing out this. We'll try to have our deterrent in a transformed the deterrent in a repellent way. So we just need more years of work, but it's possible, definitely.
It seems like if you do a whole field, it might be more of a deterrent than just one plant. On the other hand, though, a few of them get out there and they're not so selected, all of a sudden you lose the best.
Yeah, for sure. Here we have one question. Sorry, how does this compound compare to other compounds? I guess that you mean for compound other pesticide, available pesticide out in the market. Our results show that we have a comparable efficacy of our deterrent in comparison to available compounds, but we still need more work. These are just preliminary data we're collecting year after year, but we need more work for sure. But we know, we perceive that this technology can be extremely competitive to the available commercial compounds. So I hope that this was the mean of the question here. If not, please just tell me.
Is there any idea of maybe like a washing effect of rainfall?
Mm-hmm. Yeah. Thank you for the question. The question is about how terrain can impact the efficacy of this deterrent. Last year, so not summer 2021, 2020, even if the pandemic, we were able with one to perform a trial in which we had the treated berries washed by water, just to simulate, what could happen out in the field because the irrigation or because the weather. And actually, what we saw is that results were still comparable with treated berries not washed by water. I guess that the residue on berries can be still there. Of course, everything is in relation on how much water are we talking about, and if berries are really washed or just few drops of water. So it's in relation of multiple variables, but still, if we're talking about just a sprinkler irrigation system, as we simulated out in the field, we actually didn't have any negative impact on the efficacy of the deterrent. Yeah. Oh, Bashkar say thanks. So I guess that that was his question. yeah, if you don't have any further question, I guess that I can say bye-bye and see you later. Thank you.
Good morning. Can the people hear me now? Can people hear me? Is there a good way to know that? Okay. What I'd like to do today is, talk about a project on recurring blueberry shock disease. And this is a project that was written up as a one-year project with Yanis Janatakis at the University of Arkansas and myself. And we obtained additional funding from the commissions with the help of Tom Walters and with the Washington Blueberry Commission and Jason Myers with the Oregon Blueberry Commission. And I'll sort of go through what we've done and where we're at and where I think it should go. But first, a little bit of background. Growers have expressed concerns about recurring shock disease in Washington and Oregon. And when testing these plants using standard methods, we were only ever able to detect blueberry shock virus. And so that's a bit of the background. And something that'll come up a little bit later in the talk is that in 2015 to 18, Yanis and I did a national blueberry virus survey where we obtained about 2,500 blueberry samples from around the country, extracted nucleic acids, and we tested those for 13 different viruses, sort of figure out which viruses occur in what part of the country. And from that, we can give growers an idea of risks. And what we did at the time, though, is we saved those extracts and saved at minus 80 degrees with the idea that we'd be able to use them in the future if we discover new blueberry viruses. Well, the future is here. And the other thing is that I retired at the end of 2019, right about the time we found these two new viruses in blueberry. And that's why we put this proposal together with Yanis, was so that we could get something started on this recurring shock and these two new viruses, and hopefully have something in place when a new plant virologist was hired. And Dr. Dimitar Molov started in the position as my replacement in May of 2021, and is now leading this and other small fruit work at the USDA. So I'd encourage you to go pound on his door. So the sources of these two viruses that we found, there's a novel blueberry ludiovirus and a carlavirus. Initially, we found both of these viruses in a project that Yanis and I were doing to compare high throughput sequencing as a detection tool, comparing that to PCR and ELISA. And we found these two viruses in our standard positive controls that we were using, but without the HTS, the high throughput sequencing, we didn't know they were there. So once we had that, then we started looking at using high throughput sequencing to look at plants that had recurring shock symptoms in Oregon and Washington. That was funded by this project. And so we looked at 14 different plants with recurring shock symptoms, and several plants that had typical shock symptoms using HTS. And the other thing we did was we looked at plants from Michigan, actually a plant from Michigan that had bronze leaf curl disease because of work we had done with that disease earlier on, which I'll come back to that. This is an example of recurring shock in a field of Draper in Oregon. And the surprising thing about this is a very tight oval little patch like you would expect with a nematode transmitted virus. We collected nematodes in Inga's lab. We collected soil. Inga's lab did not find any Ziphinema there. We could not detect any tomato ring spot or tobacco ring spot in these plants. But with HTS, we did find that they had blueberry shock, blueberry scorch, and the ludiovirus that we had observed. And these are plants from Washington that were samples that were collected by Tom Walters. This is Draper again. And these were positive for blueberry shock and the ludiovirus in the HTS testing. Bronze leaf curl disease is a disease from Michigan that we worked with Annemiek Schilder back about 10 years ago. And we found this virus called BVA, but during that survey that we did in 2015 to 18, there's a lot of plants in the Great Lakes region that have BVA that are symptomless, telling us that bronze leaf curl disease is more than just BVA. There's something else there. And when we did HTS on the one plant we got from Michigan, it had the ludiovirus as well as BVA. So plants exhibited repeat shock symptoms in Oregon. The data showed the presence of the ludiovirus in 13 of the 14 plants that we looked at using HTS. Some of the plants were also infected with blueberry scorch, and the novel ludiovirus was not present in the 2 samples that had typical shock when we did the HTS. We only found blueberry shock in those 2 plants. And the presence of blueberry shock and the ludiovirus was confirmed by RT-PCR in 13 of the 14 plants that exhibiting the recurring shock symptoms. So we had 1 plant with recurring shock that was negative for shock and the ludio, which we can't explain. The initial survey results, I mentioned that we did this survey back in 2015-18. We had stored 2,500 samples in the freezer. And so I pulled 1,000 of those samples and sent them to Arkansas. And Yanis's group did the reverse transcription and PCR on those, testing for initially the ludiovirus. And once we had primers for the new Carlavirus, they added that to the list as well. And the primers developed for the ludiovirus was based on sequences of 7 genomes that were obtained with the HTS work. So we had 7 full-length sequences that we used to design the primers. And now at this point, over 600 of the samples have been analyzed, and the ludiovirus is widespread. But since the primers were based on 7 sequences, our guess is that the numbers we have understate what's really there, because there's a good chance we're missing some of the viruses due to diversity. Based on the sequencing we've done, virus identity can be as low as 83%. So out of 100 nucleotides, 17 could be mismatches, in which case primers may or may not work. So the occurrence of the ludiovirus, widespread. We looked at 607 samples. More than half of those have the ludiovirus. And the samples we've looked at this point are from the Northwest, Michigan, and New Jersey. Those are the areas we focused on. And in the Northwest, roughly 30% of the samples were positive for the ludiovirus. In Michigan and New Jersey, it was over 60%. So this virus is widespread. And it's probably also spreading within the regions. It's not just coming in. There may be some that came in on planting stock, but it's likely spreading within the region, because within a field, we have diverse isolates. So it's probably, if something came in on the planting stock, I would expect a single isolate. If it's moving in the field, I would expect diverse isolates to be in the same location. And this is the gel on the right. That bright band is about 1,000 bases, and we purify that, and those are the ones we sequenced. Now we have sequences for over 250 of those, and that's where we see that identity ranging from 83 to 100%. About the time, shortly after we identified this virus and had an assay, Kim Hummer at the repository was concerned that, is it in any of their material? So she identified 16 cultivars that are requested commonly and asked if we could test those for ludiovirus, and 7 of those 16 tested positive, and all were symptomless. We find this virus in a lot of plants that do not have any symptoms when it's in single infections. The other virus we found was a Carlavirus, and this is related to blueberry scorch. Down in the center of the slide, you can see the areas that this is the new Carlavirus, and the 3 closest viruses sequences that it's related to are all blueberry scorch virus that from the database, GenBank. So it's a distinct virus, but the nearest neighbor is blueberry scorch. And this is one that it gives a really weak reaction in ELISA when we do a blueberry scorch virus test. It's about 0.2, and a good positive is somewhere between 2.0 and 3.0. So in a lot of cases, this might be read as a false negative. And the PCR tests we had for scorch do not detect this virus. And in this testing we've done so far with this, it's about 500 samples. It's present in about 4% of the samples. We found it in the Northwest, we found it in Michigan, and we found it in New Jersey. Blueberry scorch virus, when we put it on blueberry scorch virus on this host, Nicotiana occidentalis, has very mild symptoms. This new virus has very severe symptoms. And another host that we work with in the greenhouse quite frequently is Chenopodium quinoa, basically lamb squirters for you that try to control weeds. And with blueberry scorch, it causes local lesions and a systemic mottling. And this new virus doesn't cause any symptoms in quinoa, but it does infect the plant. And we're in the process of purifying the virus so we can make antibodies. The antibody production, we got the purifications underway, and hopefully we'll be able to get that sent off for antibody production in the near future, and ideally have an ELISA test this summer. The novel Carlavirus identity, again, can be fairly diverse. We only have 20 sequences, and we have identities as low as 85%. Like I said, it's present in the 3 areas we looked, in about 20 out of the 500 samples we tested. So the novel ludiovirus, it's widespread. It's present in most blueberry plants exhibiting the recurring shock symptoms. It's present in the one plant of bronze leaf curl we looked at. It's widespread in the production areas we've looked at, Pacific Northwest, Michigan, and New Jersey. It's present in a significant number of symptomless cultivars at the repository and other places. It's very likely this virus now occurs wherever blueberries are grown worldwide, since it was not in any testing program, and is symptomless in many, if not all, cultivars when singly infected. And if we look at New Zealand as an example, that's probably the toughest place to ship plants into. They would not have picked this virus up because they're using all the tests that we've developed. And so they wouldn't have had a test for this virus. And that's why I suspect this virus is widespread around the world. We found it in very old germplasm and fairly recent germplasm. So it's widely distributed in the germplasm. But it should be noted that there are asymptomatic plants that did test positive for the ludiovirus and shock. So there's something else going on here that we're not aware of at this time. So we can say we do not know its role in any blueberry diseases for certain at this point. So where do we need to go with this work? We have the 250 partial sequences obtained from the initial survey, and Yanis is in the process of developing primers based on those 250 sequences, so we should have a more reliable test, and is doing the same thing with the Carlavirus, though we only have 20 sequences, and that's being funded by the Farm Bill. And then there's a whole list of things that should be done. The cultivar differences to infection by these two viruses. How do different cultivars respond to these viruses? Blueberry shock together with the ludio, blueberry scorch with the ludio, the new Carla with the ludio. Start looking at these combinations to see where this recurring shock might become, what might be causing the recurring shock. Identify the vectors for ludiovirus and Carla. That TBF means to be funded. So you can put your hand up and pick one of these items to fund. Virus interactions and vector management. So once we know what the vectors are, how do we manage that? Examine the recurring shock disease using the different primers. Examine symptomatic plants that are negative for shock and scorch and luteal. Again, this would be some more HTS work. Based on the work that we've done getting samples from Jason Myers at Crop Pierbolt and Tom Walters at Walters Ag Research, somewhere between 10 and 20% of the plants that are showing what they identified as recurring shock were testing negative for blueberry shock and the luteal and scorch. And the group up in British Columbia, Peter Ellis at Phytodiagnostics, been doing testing up there, and they're finding fairly similar percentages. So there's a significant number of plants showing this recurring shock that are negative for these three viruses. So then the question becomes, is there a new strain of blueberry shock that may be more severe and capable of causing recurring shock symptoms in a single infection? And if there's a new variant, our new Delta or Omicron variant of shock that we might be missing with the ELISA test and with the PCR test. Again, that's where we need to do HTS, high throughput sequencing, because that should be able to pick up viruses that we don't know about. That's the best way to look for those now. And again, that's something that to be funded. And long term, look at sources of resistance, tolerance, and field resistance. Here I'll say continue to be funded because the commissions have funded CHAD to look at plants slow to get shock for a number of years. And I think Claire and Demeter may be interested in continuing that. So it was a one-year project with the idea of putting something in as a stopgap until we had a replacement. And I think working with Yanis, that was a very productive project. But it was a one-year project with the idea that my replacement, now Demeter, and his colleagues will continue this work. And talking with Demeter, he's interested in continuing this work to really get a handle on what is causing this recurring shock and how to manage it. So let's see. I do want to acknowledge the funding sources, this project from the Northwest Center for Small Fruit Research. The Washington Blueberry Commissions helped support some of the PCR testing and supported Tom Walters and Jason Myers' time as far as doing the collection. And it's great to have people in the field that know the fields, know the growers, and can collect the samples. Farm Bill funding. The Farm Bill funded the diversity studies with the luteal virus, and National Clean Plant Network funded the comparison of high throughput sequencing with the traditional testing methods, and that's how we initially identified these two viruses. So funding from a number of sources. And then I am mostly retired, so trying to find something else to work on. The meaning of life can be discovered in a fine bottle of wine, and I still haven't found that bottle. And with that, I would like to wish everybody a happy holiday season and a great 2022 and take questions. So any questions? The plants with shock produce fruit. Do we know what the fruit quality is like? We haven't really looked at that. It's something that might be a good project to look at, regardless of recurring shock or plain shock. With normal shock, the plants recover. We've looked at yield. We've not looked at fruit quality, but it's something that should be considered. In the cases with the recurring shock, we tend not to get very much fruit because with the shock symptoms, most of the flowers become necrotic and there's very little fruit produced on that plant. And normally with shock, that happens 1 year and then plants recover. But with this recurring shock, it's year after year. So it's becoming a non-productive bush. Luteovirus and fruit quality. We haven't looked at that. The luteovirus has just been recently discovered, but we have a lot of symptomless bushes that have the luteovirus. So that's something that could be looked at. And in that case, there should be plenty of fruit to do that. And again, it's one of those things that is there going to be a cultivar difference? And if you start looking at different cultivars, it gets to be a pretty huge study. But if the fruit analysis is fairly straightforward, you could do a lot of samples. I think in the berries, we generally have not looked at fruit quality in terms of, I mean, with rubus, we have crumbly fruit that becomes unmarketable. But if the plant's infected and symptomless, we haven't looked at fruit quality. I know in grapes that's been much more of an issue. Does luteovirus change the chemistry of the fruit? Yeah, we do not know if these viruses are affecting fruit chemistry in any way. We just haven't looked at it, but it might be something to set. Somebody could set that experiment up and look at it, add that to the list of next steps. They are looking at the luteovirus up in Canada as well. Peter Ellis is doing that testing. And again, they're finding a similar thing where they have symptomatic plants that are testing negative for both. And I think if we do HTS on those plants, and we had the one plant that was negative, but you wonder about just poor DNA or nucleic acid quality maybe. But if we start look at 10 plants and they're all negative for any known virus, then it's probably caused by something non-viral. Maybe there's something else that can cause that type of reaction. Any other questions? Well, I would like to thank the organizers for having this be a hybrid meeting because we have plans to visit with grandkids this Christmas, and we're not doing much interaction with other people between now and then since the grandkids aren't vaccinated. So this gave me the opportunity to present the information without potentially becoming exposed. So thank you for that. How easy to eliminate these viruses? The Ludio virus should be fairly easy to eliminate because it's phloem limited, which means that it should not get into the meristem because the meristematic tissue isn't developed in the phloem yet. And doing meristem propagation, they've been able to get rid of this. So I think the Ludio virus is easy to get rid of. The Carla will be a little bit more challenging, but it's doable. And then with all these new viruses and problems occurring, is there any thought developing vaccinations for new plants? Vaccination for viruses is at this point is RNAi, and it's technically it's very simple to do. Politically, and it's very difficult to use that technology and be able to sell the fruit as freely as we can now. There'll be a lot of places that would resist purchasing that fruit. We don't have a vaccine. They don't have an immune system so that they can clear the virus with antibodies. So that RNAi technology at this point is somewhat challenging. It'd be considered GMO, and CRISPR technology. We don't know if we'd be able to do that. That we have a gene we could put in to provide virus resistance. Yeah, the GMO thing is just politically, it's a tough sell at this point yet. I think if the CRISPR technology becomes useful, that would be the way to go, where you can modify a gene rather than put something new in. Okay. Anything else? And if not, I think I will stop sharing.
Good morning, and thank you for your patience, everyone. I'm sorry that my presentation is not showing at the moment, but I'll just talk you through my update. Our project is about black raspberry necrosis virus resistance. And this is a project undertaken by myself, Nala Basil, a USDA ARS research geneticist, and Christina Mulch, a master's student who will be defending her thesis this Friday. Our goal is to find genes that confer resistance to black raspberry necrosis virus, which is vectored by the aphid Amphorophica agathonica. We are building on previous genetic mapping work that was done by Jill Bushakra, also at the USDA ARS. Her work was with molecular markers and genetic maps, and she was able to narrow down the resistance region in the black raspberry genome to a region on chromosome 6, but wasn't able to narrow down that region to specific genes contained in that region of the chromosome in the black raspberry genome. And so our work is to refine and discover genes in that region and in other regions in the genome where black raspberry has genes that can resist the virus and the aphid that vectors it. For this work, we're using messenger RNA, and we're exploring gene expression. And unfortunately, I can't show you in my presentation where in the scheme of things messenger RNA is, so I'll just talk you through it. When genes are expressed, an intermediate molecule is produced called messenger RNA, and that's on the way to being translated into a protein that does various function in the cell. And so we're looking at gene expression and thinking about ultimately what protein products of genes are used in a resistance response by the plant. And it turns out that there are different genes in different plants that appear to be responsible for conferring this resistance to the aphid and the virus that it carries. For the work that Christina is doing, she's ultimately comparing she's making different pairwise comparisons. She's comparing plants with and without aphid inoculation. She's comparing resistant plants to susceptible plants. And she's comparing plants that are harboring different genetic sources of resistance. And so it's very important that there are different genetic sources of resistance in different black raspberry plants. That's really good because that means that if a particular resistance gene is overcome by the virus and the aphid, then there are other resources that can be used. There are alternative genes, in other words, that can be employed in a breeding program to confer resistance. Ultimately, we want to develop and test molecular markers for these different genes that we're finding. So Christina has 3 different genetic sources of resistance derived from 3 different geographic regions. There's an Ontario source, a Maine source, and a Michigan source. And different segregating populations have been developed for each of those 3 sources. And those are the ones for which Christina has been exploring gene expression profiles. And it turns out that each of the 3 segregating populations is segregating 1-to-1 for resistance versus susceptibility. So about half of the plants in each population are resistant to BRNV, and about half are susceptible. And she's identified sets of genes that are highly expressed in resistant plants, but not in susceptible plants for each of the 3 populations. And it turns out that these genes aren't the same genes, which we expected. We expected to see some genes that were different and some genes that are the same, but in a lot of these comparisons, we saw more genes that were unique to one population or shared by 2 populations, but not by all 3. So we've been pawing through these lists of differentially expressed genes in these populations and trying to figure out what they are. And here we can take advantage of genes that have already been found to be resistance genes for plants for various pests and diseases. And so we can use that knowledge and we can look for similar genes in black raspberry and see if any of them are indeed correlated with aphid resistance. And so on the last slide of my presentation, I have a short list of those showing that there are gene IDs for annotated genes in the black raspberry genome. And they are similar to known resistance genes from other plants. And so those are our top candidates for molecular marker development. And we hope to continue on with that work and take the next step to test and refine molecular markers for BRNV resistance in a wider swath of black raspberry germplasm, and ultimately deploy markers in breeding for marker-assisted selection. And with that, I'm happy to take any questions if time allows. I'm not hearing any, I'm not seeing any in the chat, so I'll stay on for a while, but I'll go on mute and see if any show up. Thank you for your attention.
So Mary, I see a question from you saying that, yes, there was technical difficulties. Yes, it will be repeated later today. It will be repeated at 4 p.m. today. And it looks like Kelly actually sorted out a way to make it happen for this afternoon at 4 p.m. So sorry about all that. And we will be moving to the next speaker shortly. Thank you.
All right. Well, good morning, everybody. So today I'm going to talk about a project that was funded for 3 years by the Northwest Center for Small Fruits Research on irrigation strategies for optimizing water use efficiency and improving fruit quality and cold hardiness in trailing blackberries. Collaborators on this project are my grad student, Jess Carroll, who's been collecting most of the data in this project this past year. Bernadine Strick and Amanda Davis, who were instrumental in getting the field planted, and they're actually using most of the field for a study that they're talking about, that they're presenting on different management practices for Columbia star blackberry. And then my technician, Scott Orr, who was really the guy who put the lysimeters that I'm going to talk about together and got all the electronics and everything else going. Without him, obviously, I couldn't have done this project. Let's see. Okay, that doesn't work. What he told me does not work. Yeah, if you can grab him, I'll try. He said click on this. There we go. Got it. Okay, so where this project started from was trying to get a better idea of how much water blackberry plants required. I did a lot of this work when I was with the USDA down in California at the Water Management Research Unit in Fresno, California, where we put in these large weighing lysimeters to estimate what were the water requirements in vegetable crops. Well, ever since I moved here in 2003, I've wanted to do something similar in berry crops, but these projects are expensive. The one I did in California, that was a $500,000 project. So we had to come up with a more cost-effective way of doing this. But why is it this important? Well, obviously, you need to know how much water your plants need if you're going to irrigate. So one option that growers do have currently is to go to these weather-based weather network sites. Here it's called Agromet, or there's AgWeatherNet up in Washington, where growers can go and actually get information on what the estimated water requirements are for various crops. So here is a summary from 2001 that I downloaded yesterday, from June 22nd to July 9th. And you can see it has the date. It has ETR, which is called reference evapotranspiration. And what that means, that's the amount of water that's estimated for an alfalfa crop at a given time. So depending on what the weather conditions is, the hotter it is, the drier it is, the more water an alfalfa crop is going to use. And this is all based on calculations from a formula that was developed back in the 70s to estimate, what is the potential evapotranspiration in the field. Now there's two different reference ETs that we use. One is ETO, which is the more accepted standard, and it's based on grass, or alfalfa, which is signified as ETR, and this is primarily what Agromet uses. So that's why I have this up here. And then next to that is the estimated water used for the 3 berry crops that's produced here in Oregon: strawberries, trailing berries, so that encompasses both blackberries and raspberries. And then for blueberry, you'll notice I highlighted in red 3 dates, June 26th to June 28th, where you can see that the reference ET, as well as the evapotranspiration or estimated evapotranspiration for strawberry, berry, trailing berries and blueberries were quite high. Well, that was the heat dome event. And you can see on June 29th, which was the worst day, that's where it got up to about 116 degrees in Aurora, the reference ET was about a ½ inch of water per day, which is much higher than what we'll typically see, particularly in June. And consequently, that's why we saw the plants struggle so much this past year. And you can see for trailing berries, for blackberries and raspberries, it was also around a ½ inch of water on that day. Now, if you look after that, you can see that the water use was anywhere from 0.13 to 0.28 inches per day. And that's more normal than what we'd expect for this time of year. So where do they get that information? Is, well, one is they get the estimate of ETR from the weather data. So they have weather stations located out throughout the state collecting weather data. So you would go to that site that's closest to your field, download that information, and then they multiply that ETR by a crop coefficient, because obviously a blackberry plant is not going to be using the same amount of water as an alfalfa field. So you have to correct for that. And so what you do is you simply multiply ETR by that crop coefficient to calculate what the estimated ET is for a given crop. So here is a curve that they use for trailing berries, and it goes from 0 to 200% growth stage. It's kind of weird, but basically what they do is they sort of divide the curve up from bud break to full cover, which is 0 to 100% growth stage, and then full cover to dormancy. So as you go through the course of the growing season, you get an estimate of what the crop ET is for that particular crop. That's great. And there's a lot of science behind this. And there are some very good curves for certain crops, like wine grapes, for example. There's been some very good work that's been done to develop crop coefficient curves for wine grapes. However, for berry crops, such as blackberries, this is all theoretical. So this curve is just one size fits all for pretty much all the berry crops. And obviously, the way a raspberry grows isn't like the way a blackberry grows, and the way a blackberry grows is nowhere like how a blueberry would grow. And this all affects that crop coefficient, basically that architecture of that canopy, and how that affects the physics of water use in that given crop. So what we wanted to do was actually to develop a real crop coefficient curve for blackberry. So one of the best ways to do that is to use what's called the precision weighing lysimeters. And this is what I was talking about where when I was in California, we had installed some of these weighing lysimeters. And the project was quite expensive. Each lysimeter cost about $150,000. And then include that with all your tech labor and so forth. It was a very expensive project. But in that case, those lysimeters were larger, very sensitive. So we had to scale it down and come up with a lysimeter that we could actually fit within the budget with what we could work with in a berry crop. And all the way precision weighing lysimeter is basically a soil tank sitting on a scale. So what you do is you have an outer tank or an outer chamber and an inner tank. And on the bottom of that outer chamber are load cells that basically is like a scale, so that when you have the inner tank sitting on it, fill it with soil, and then have a plant growing in it, it's going to measure any weight changes over the course of the day. So if you have a blackberry plant growing on this lysimeter, as it transpires and as evaporation occurs, there's going to be a weight change that happens. And if you irrigate, of course, the weight will go up, and then it'll use that water. Well, we can, using data loggers, collect data. Basically, we're collecting data every minute and then taking 15-minute averages over the course of the day to see exactly how much water the plant growing in the lysimeter is using. So on the top here, you see a picture of Scott here downloading data from the data logger from one of those lysimeters where we have the blackberry plant growing. And this was the first year. The other thing we're doing is we have a drone where we're collecting remote images. So here's an example of NDVI, so that we can relate how big the plants are to how much water they're using. So obviously a smaller plant's going to use less water than a bigger plant, but we need to know that relationship so that so when we make predictions, it's not just the given date of a year and the weather, but also what stage of development the plants is, because obviously a plant during the first growing season is going to be very different than once it's mature in year 3. So what I have on the bottom right here is an actual set of data that we collected during that first year with the lysimeters versus what we downloaded from the Agromet station at the Northwest Center where the lysimeters were located. So we have the evapotranspiration, the reference ET, I have ETO, but this is actually ETR, in the white dots, and then ETC in the black dots. So you can see that basically every time that ETR drops, so does ETC in the lysimeter. So, and that's because of the weather conditions. So as it's cooler on a given date, ET is going to go down, and when it's hotter on a given date, it's going to go up. So there's a ratio between the two. So if we take ETC and divide it by ETR, that gives us a crop coefficient at that particular stage of development. And so if we do that over the course of the growing season, we then get our crop coefficient curve. And now the curve, the first year is going to look very different than what that curve is that you're going to get from Agromet or AgWeatherNet, because this is an immature crop. So basically what's going to happen, it's just going to continue to increase as that canopy continues to increase in size during that first year. You could see too, if you look on the top left, so here is an actual curve of the crop coefficients that was collected using the lysimeters. And it's not quite a straight line, because at some point you have to train those primocanes onto the trellis wire. And when you do that, that actually changes how the plant is intercepting light. So consequently, it's using less water during that point. So it kind of drops a little bit, but then as that canopy builds up again, it starts using more water and kind of gets back in line to where it was, would have gone had it not been tied up. This past year was our baby crop year. So now we do have a complete set curve, or we will. We're still analyzing the data from this past year, but we'll have a curve for this entire year. The plants did quite well. You can see here in that center photograph, a picture taken from the harvester, and you can see that the uniformity of the plants in the row are pretty much the same in the lysimeter as outside the lysimeter. And that's very important for lysimeter research, that the crop in the lysimeter has to grow just like the crop outside of the lysimeter, because what could happen is, say, if you had more growth in the lysimeter than outside the lysimeter, you get what's called an oasis effect, and your plant will actually be using a lot more water than it would otherwise. Or obviously, if it's not growing as well, it's not going to give you a realistic value of water use. Now, unfortunately, much like the growers, they had this horrible heat dome event in late June that cost them a lot of money and lost a lot of fruit in the process. Well, we also had problems as well. And you can see here on July 7th, there's my graduate student, Jess Carroll, who's giving a talk about the lysimeters. And you can see on the plant that all the heat damage that occurred during that time. And we lost about 70% of the crop during that time. So what's going to be cool, though, is that we will now see what effect that heat dome event had on water use in blackberries, because the thinking is that blackberry plants shut down. Well, we're going to be able to see, did they actually shut down in terms of their water use during that time? Or were they just putting out a lot of water, but it still wasn't enough, just wasn't enough to prevent the damage? Some questions we're going to ask, obviously, as this project continues. We're going to keep this in for at least another 2 or 3 years as the planting is mature, is what is the crop coefficients in a mature planting, and we'll be in our mature planting this stage this next coming year. We also have 2 different lysimeters, so we can do 2 different types of training systems. One is the traditional training systems where you just train your canes up in August. And the other is what Amanda and Bernadine were looking at is new over old. So basically just leaving the old floor canes up and then training the new primocanes on top of that. And we can see what effect does that have on water use. The other thing we can do once we test that is the next year we can look at water stress. We can have one lysimeter where we're going to irrigate the plants fully, and we have another lysimeter where we actually reduce the amount of irrigation and see what impacts that has on the water requirements of the plants and kind of get an estimate. So if you don't have enough water, and you cut back, how much can you cut back by and still not impact productivity in those plants? With our drones, we have different types of cameras that we're using. We have a near infrared camera, where that one photo I showed you, where you get things like NDVI and so forth. But we also have a thermal imaging camera that allows us to look at the temperature of the plants. And why that's useful is that when a plant is stressed, it doesn't transpire as much water. It closes its stomates. And when that happens, the leaf temperature goes up. So you can actually see when a plant is stressed using these thermal imaging cameras. So what we're doing is we're developing what's called a crop water stress index, where we can actually use these drones to estimate what is the water status of plants. We can estimate what the leaf water potential are simply by flying over a field. So that way, if a grower is not irrigating enough, or if they have some problem with their irrigation system, you could fly over the drone and quickly see that there's water stress in the plants, and we could see exactly how stressed they are. Obviously, once we get the complete set of crop coefficient curves, the next step then would be to integrate that information into these sites like Agromet and AgWeatherNet. so that we now have a more accurate estimate of what the water requirements are. So if growers have a better estimate of what the water requirements are, then obviously they're not going to stress their plants. They're going to maximize growth and maximize production. So clearly there's going to be an economic benefit to that. The other thing that we have is this tool called Irrigation Scheduler, which is a mobile app that was developed by Troy Peters at Washington State University. I don't know if anybody's tried this app, but it's a really cool app. You kind of enter the parameters of your field, what size it is, your plant spacing, what your irrigation system is, and it spits out each day what the estimated water requirements are. Well, we'll be able to integrate that into not only to Agromet, but into this app so that we get a more accurate estimate of the water requirements in a given field. The other thing we're doing now that we're getting into the mature stage is we had some extra rows that we were going to look at water deficits, because I know growers, some growers will irrigate the whole season. Some growers don't irrigate at all. Rare, but that does occur. Some will reduce irrigation and just sort of give enough to get the plants through, and some will shut off irrigation after harvest. And we want to look at these different treatments. So we're going to have 3 different treatments out there. We're going to have full irrigation, reduced irrigation, where we'll give about half as much water as what full irrigation would be based on the lysimeters, and then no irrigation after harvest from August, September. And we had with Dr. Strick and I and a few others had done a study a few years back in organic blackberries where we found that actually cutting off irrigation after harvest was beneficial because it increased the hardiness in the plants. So they were more hardy going through the winter. And that's what a lot of growers thought, and we indeed did show that. And we estimated in that particular project that saves 67,000 gallons per acre by doing this. So you need, obviously, you're going to have a benefit by using less irrigation water and paying for that, the cost of pumping that water and so forth. But anyways, the results of no post-harvest irrigation in that study was it obviously reduces weeds because you don't have any water for them. Had little to no effect on fruit production. It reduced winter injury, and it saved water. So we're going to see, we're going to confirm this in this particular study and see if that's the case. So that's pretty much where we're at in this particular project, and be happy to take any questions at this point.
I'm curious if you have all your growers lined up yet for trials over the next few years, or if you're open to others potentially.
So you ask if we have our trials open to other growers. What do you mean in that regard?
Well, sometimes researchers have, already talked with growers and have people lined up, and other times there's kind of an open call to see who might fit the experiments best.
Right. Yeah. At this point, yeah, we're not to the stage where we're taking it to grower fields and actually validating that. So no, we don't have that set up, but that is something we do anticipate once we get, the next couple of years of data collected. Yeah. Yes.
How did you schedule irrigation to ensure that I almost said vines, you can tell what crop I work with, to ensure the plants were well watered?
Right. So
we're not stressed in the lesson.
Okay. So the question is, how did I ensure that we were fully irrigating the plants, that they weren't stressed throughout the growing season? Well, that's the beauty of the lysimeters is that it tells you exactly each day how much water the plants use. So we know what the maximum water holding capacity of the lysimeters. We look at the weight change over the course of the day, and then at the end of the day, that tells us exactly how much water that plant used. So then we just irrigate with that amount of water the following day. Every day or two, depending on the weather conditions. Yeah. So, and we may set it up differently now that we got mature plants. We didn't want to,
Is this manual or is this auto?
Oh, it's all automatic. It's all with data loggers.
Weight change the lysimeter, the pump comes on, irrigates,
Correct.
and then it goes to fuel capacity.
Correct, yes.
And then it just
Yeah.
cycles.
Correct. And we have a drainage system on the bottom too. So that when it rains and it becomes oversaturated, it goes into that drain. And then we have a pump that kicks on automatically and takes the water out of it that way
So you
too.
feel pretty confident that they're at fuel capacity as much as possible.
Well, we do now. I think, there were some, there's some, getting used to it kind of a thing. And, Jess kind of learning how to use this and so forth. But I think going into this next season, we have a very good idea of how to do it now. So I think, the data is only going to get better as we move on. And that, now that we're at the mature stage, that's where it's really important.
And so you're doing flyovers with drones looking from the top down. Have you tried to do any measurements of like canopy shaded area just because it's kind of like an easy way that a grower might do it to figure out what their own KC could be. It's been done in wine grapes.
Right. Yeah. So the question was, we'd use the drone, look over the top of the plants. And so have we looked at shaded area? And that's, there's work done. When I was in California, there were guys doing that, like Larry Williams did it in table grapes and extrapolated that to wine grapes. And then there was some work also done in peach trees, same thing, trying to get an easy way for growers doing it. Well, using the images from the drone gives you pretty much exactly the same data as if we were to measure the shaded area at midday. So I think, then there's been studies that actually have compared the two and they're identical. So we don't do that, but I'm very confident that, had we just been measuring shaded area versus over the top, but that's really difficult to do in a plant like a trailing blackberry because there's so many canes there.
It's like on the ground.
Yeah. So I
It's a good relationship between what you get from the top of the drone and your actual measured KC value.
Yes. Yeah. You tend to get a very good linear relationship with that. And we are finding that is the case. Yeah. And that's nice. I think probably, for blackberries, blueberries is going to be a little bit easier because we're now starting a very similar project in blueberries. In blackberries, I think it's going to be more of a visual estimate, kind of looking at it and saying, yeah, that looks like about 30% kind of a thing. Because I really don't see anybody going out underneath the blackberry plant and trying to, map out what the canopy area is underneath that.
We can use a septometer.
Yeah, that's even difficult because there's just so many canes. If you look at the photograph here, you can see all those canes.
You can make a lot of measurements.
Yeah. I mean, it could be done, but I don't see a grower,
I'm just wondering if
wanting to
the
mess with it.
relationship between this like canopy cover, shaded area, whatever you want to call it, and your KC has a similar slope to what we find in grape.
Right.
Because actually, I think the slope in like grape and in peach and some other crops are all fairly similar.
They are. Yeah.
So it's kind of this interesting, like highly conserved number.
It is, and our work in vegetable crops pretty much showed the same thing. They all kind of fit on that line. Here's what I'm thinking. What I'm thinking is growers, rather than having to actually go out and be measuring shaded area, eventually everybody's going to have drones.
Oh, yeah.
And what we're trying to do is, we're working on that technology so that growers can actually get site-specific estimate of crop ET based on canopy covers with the drone. So if we can get to that level, then doing all this other stuff is going to be outdated.
Yeah.
Good questions. Thanks. Anybody else? Yeah.
I know you just said two.
Correct. Yeah. Well, I mean, each one's $10,000. So
I know.
yeah, right. Plus data loggers and all that other stuff. So
I mean, we all agree you should have more money, but how much variability do you see?
between the two,
Yeah.
they're almost identical.
Yeah.
We see almost no variability between the two, which is great. And if you look at, there's been hundreds of lysimeter studies done all over the world, and it's very rare that anybody replicates that because it just is so expensive and time consuming to do that. So having two is great. With blueberries, we're actually working on having more lysimeters. I don't know if we'll actually be able to replicate it with 3, per cultivar kind of a thing, because with blueberries, we're looking at early, mid, and late season cultivars because we know the water use is so different in the different ripening periods. But so we'll have more lysimeters for them and have that at least replicated twice. The goal is 3 times. I don't know if I'll be able to scrape enough money for that because price has gone up since too because of the price of steel. Now we're talking $12,000 just for the steel component of the lysimeters. So yeah, they're getting pricey. Yes, Andrea.
Do you think it'd be do you think growers would be likely to stop irrigating post-harvest Especially with like the rising temperatures and such. Do you think it'd be easy to convince them?
So the question is, will it be easy to convince growers to stop irrigating post-harvest? What do you think? Do you think it'll be easy to convince growers? I think a lot of them do that already. So I think, you know, being able to tell them whether that's a good thing or not is going to be very useful. So yeah, I don't anticipate that being an issue. I think it might be harder to tell them that you should be irrigating post-harvest, in fact. So yeah. And I don't know if I can access the online. Oh, yes. Okay. Okay, so here's a question from Dr. Bandata. What tools do growers use to schedule irrigation? Well, that's a good question. I think, you know, in a lot of cases, growers just use historical values on how much irrigation to apply. We know that blackberry plants use about an inch to 2 inches of water per week over the course of the summer. So they'll typically, if it's cooler, they'll add about an inch, and then they might go 2 inches. Growers that are more sophisticated will actually look at the soil water content, or, you know, they'll have some sort of measurement of that, and they'll use that for their irrigation scheduling. And that's great, particularly if you have very uniform soil in your field. However, if your field's not uniform, it's difficult to know where to put those sensors and to get an accurate estimate of when your plants need to irrigate, because obviously you'll be irrigating the ones where your sensors are, but there could be other spots in the field that's either getting too much or not enough water. And then some growers are actually looking at this data from Agromet and using that as an estimate. But again, this is just an estimate, and it doesn't account for, you know, the stage of your field. You know, if it's a young planting, it doesn't account for cultivar. It doesn't account for you know, a lot of different things. If you have any stress going on, for, you know, say if they're nutrient deficient, it's not going to be the same as a very good growing plant. So so there's different tools available. Growers are, you know, whoever I talk to, you know, each one will have a different answer to that question. And let's see, and then there's another question that says, are there any plant indicators for scheduling irrigation? Yeah, and that's another good question. And the answer to that is no. Currently, there's no plant indicators that we can use for scheduling irrigation. I know, for example, in wine grapes, they look at water potential for scheduling irrigation. Great tool, particularly if you want to water stress your plants. We don't do that in Cambridge. I mean, we do take water potential measurements, but we don't schedule irrigation based on that, or stomatal conductance. We don't do anything like that. And that's why we're trying to, we're hopefully using this remote imaging technology, using this thermal imaging, for example, developing crop water stress index, that we can use this to help growers for scheduling irrigation more site specifically. So that's kind of, you know, another direction we're working. We'll see if I can get all this done in the next 7-8 years before I retire, but no, there you have it. And I think that's it. So any other questions? Yes.
In your figure on the upper left, you have a percent ground cover, getting back to his question.
Mm-hmm.
Is that based, that's not based on the blackberry canopy, is it?
Yes, it is. Yeah, during establishment.
Okay.
Yeah. So basically during that first year. So there's very little cover if you look at the whole area of the field during establishment, you know, because you know, you can identify individual plants. And that number was taken from those drone images that I showed in the previous slide.
Oh, that ground cover percent from the drone.
Yeah. Mm-hmm. Okay. All right. Well, thank you.
Sorry, one sec. We can't hear you. We can see everything you're doing, but we can't hear you yourself. We still can't hear you. If you could check the chat real quick.
Okay, how about now? Can you hear me now? Okay,
Yep, you're good to go. Go for it.
good. I will resume. Then was anything audible at all, or should I start right at the very beginning?
No, we could see the stuff, but we couldn't hear anything you were saying. So if you want to start over real quick.
Oh, my. Okay, good. I'll start over now. Okay. Hello, everybody. I'm here to talk about our project in black raspberry necrosis virus resistance. We're looking for genes associated with this virus resistance, abbreviated BRNV, in black raspberry. And this virus is vectored by large raspberry aphid, Amphirophora agathonica. This work is building on previous research that was initiated by Nahala Basil and Jill Bushakra at the USDA ARS. And the current work is building on that work and also hoping to expand it to look for resistance genes in black raspberry germplasm and breeding material. This work's being conducted by myself and by Nala Basil, the research geneticist at the USDA ARS, and master's student Christina Mulch, who will be defending her thesis on Friday. The previous work was done with genetic linkage mapping, and a locus was identified in the black raspberry genome that was associated with aphid resistance, but the work did not continue to narrow down that region to individual genes that were associated with that resistance. And so this work is expanding on that previous work and using a different approach, using mRNA and gene expression profiles to look for differences in plants with before and after aphid inoculation, in resistant plants versus susceptible plants, and in plants harboring different genetic sources of resistance. For those of you who aren't thinking about plant genetics all the time like we are, I have a figure here on the right showing the central dogma of genetics and mRNA's place in the scheme. When genes are expressed, an mRNA intermediate molecule is made from an expressed gene, and it's on its way then to be translated into protein that will do some job within a cell. And so ultimately, we're looking for genes that encode proteins that are involved in different aspects of plant defense. So Christina is comparing in a pairwise fashion these gene expression profiles to see if we can find genes that are involved in aphid resistance and virus resistance ultimately to target with molecular markers that can ultimately be used in breeding. For this work, she is using 3 different segregating populations that represent 3 different geographic BRNV resistance sources, one from Ontario, one from Maine, and one from Michigan. You can see in this pedigree figure previous crosses that were made using these sources of resistance. And ultimately, the plants that are starred in this figure were used to make new crosses to a susceptible individual that is not being shown in this figure in order to produce new segregating populations designated on the right here by the numbers 5306 for the Michigan source, 5291 for the Maine source, and 5296 for the Ontario source. Christina screened these individuals in these populations by inoculating them with aphids and determined how many were resistant and how many were susceptible in each population. And as you can see, there are roughly even numbers of resistant and susceptible individuals within each population. So then she was able to proceed to make the pairwise comparisons, resistant versus susceptible, pre-inoculated versus post-inoculated. And also, at the same time, comparing profiles of expression from the different sources of resistance. And this is a snapshot of part of her results. Ultimately, she's been able to identify sets of genes that are differentially expressed in these different pairwise comparisons. And in this case, we're looking at sets of genes that are highly expressed in resistant plants, but not in susceptible plants, and are also unique to each source of resistance. So as you can see in the Venn diagram, none of these genes are shared by all three populations in terms of being differentially expressed in all three populations. Some of them are shared between two of the three, but none are shared among all three. And so we're looking for genes that are similar to genes that are already known from work in other plants to have something to do with plant defense or disease resistance. And here's a little snapshot of some of the genes that came to the top as being the most interesting for us. And you can see that the gene descriptions and the functional categories indicate that they're known or there is evidence in other plants that they have something to do with plant defense, stress response, and that sort of function. And so these will now be targets for molecular marker development in the current populations that Christina has been working with and broadly in more black raspberry germplasm. And with that, I'll close and see if there's time for any questions. Thank you for listening. Okay, I see the question from Michael. Were any of the differentially expressed genes in the region of linkage group 6 that resistance has been mapped to? Christina has found the closest genes in that region of linkage group 6, and we're still analyzing them right now. But yes, there is a list of top candidates. Okay, I'm not seeing any more questions come into the chat, so I think I can stop sharing my screen at this point, and thank you again for your time.
Okay, I guess I'll go ahead and get started. Is this microphone working?
We can hear you here, it's just, you need, I guess, for them.
Yeah, I guess so. Okay. So today I'm going to talk about a project that was funded for 3 years by the Northwest Center for Small Fruits Research on irrigation strategies for optimizing irrigation water use and improving fruit quality and cold hardiness in trailing blackberries. Collaborators on this project are Jess Carroll, who's a PhD student who's working with me, Bernadine Strick and Amanda Davis, who were instrumental in helping to get the field plot set up. And in fact, most of the field is what you might have seen Amanda's talk earlier on looking at different training strategies and so forth for trailing blackberries. And Scott Orr, my technician, who's basically the hands and the brains behind all this and got everything set up and got it working and so forth. So the way this started originally is just, trying to answer the question, how much water do berry plants need here in the Pacific Northwest? And in this case, trailing blackberries. How much water do they need? I mean, currently we can get some information off of our agricultural weather sites like Agromet and AgWeatherNet, where we can download this information. And this consists of reference ET, which is basically the potential evapotranspiration based on weather conditions. And these values come from models that were developed in the 70s that basically you put in things like solar radiation and temperature and so forth, and it can calculate what the evapotranspiration is, either of an alfalfa field, which is ETR, or of a grass field, which is ETO. Now, Agromet, they use ETR, even though ETO is more of the standard for reference ET, they still do use the ETR value, and that's how they calculate their water use for other crops. And the way you do that is in order to calculate how much your crop is using relative to an alfalfa field, you have to multiply it by a crop coefficient. So for example, here's the crop coefficient curve that they use for trailing berries. So that includes both raspberries and blackberries. So one size fits all. And you can see the growth stage goes from 0 to 200%, whatever the heck that means. But basically what they do is they kind of divide it 0 to 100% growth stage is going from bud break to full cover. And then 100 to 200 is full cover to dormancy. So you have to know the date of bud break, and then it kind of figures how long that season is and plugs in those numbers. So it'll get the ETR from the weather stations data, calculate ETR, multiplies it by the crop coefficient at that stage of development on that date to calculate what the evapotranspiration of the crop. So here is an example of some data that I pulled up yesterday from this past summer, going from June 22nd to July 9th. And you can see, for example, in June 22nd, the reference ET was 0.23. Next to that is the reference the ET for strawberry, estimated ET. Trailing berries is 0.21, and then blueberries is 0.22. You'll also notice that I highlighted June 26th to June 28th. And that was that horrible heat dome event that we had in late June. And you could see during that period how much water use had increased during that period. So for example, on June 28th, the estimated reference ET was over a ½ inch of water. And if you look at trailing berries, it's almost a ½ inch of water. Compare that to other dates, you can see that was much higher than usual. And this pretty much explains why our crops could not keep up and why we had so much damage, because this ET was so high on those dates that the plants just couldn't simply take up enough water during that period. So one of the best ways to answer, okay, this is a theoretical curve. I'm not even sure where this curve came from. I mean, there have been some actual curves developed for other crops. For example, in table grapes, there's been a nice curve that's been developed. In peaches, there's a nice curve. Of course, there hasn't been anything developed for trailing berries up to this point. And one of the best ways to develop these curves is to use what's called a precision weighing lysimeters. Now, I did a fair amount of this work when I was in Fresno, California, down at the USDA there, where we installed these large weighing lysimeters, 25-ton lysimeters, which are basically soil tanks that are sitting on a scale connected to a data logger, and you're constantly getting data of the weight change over the course of the day. Now, those lysimeters, they at that time, back in 2000, cost $50,000 each to install. You know, that's a pretty big price tag for berry crop research. Down in California, they could afford those sort of price tags, but up here, that's difficult. And I also had 3 different technicians helping me out on this project. So I've wanted to do this for years, but the problem was, well, one, I wasn't going to get this much money for a project like that, like I did in California, but I knew there was other types of lysimeters out there. So we did develop a simpler form. And the other is I needed a technician who was capable of doing this kind of work. And I was fortunate enough to hire Scott a few years ago, who's able to do these types of measurements and have these sort of instruments built and design them and so forth. So what we did is we had 2 of these lysimeters constructed. And basically what they consist of is an outer chamber constructed from 3/8 inch steel. And on the bottom of that are 4 load cells in which we place the inner tank. So the inner tank sitting on top of the load cells. We connect the load cells to the data logger so we can continuously look at weight changes, fill the soil tank up, plant our plants in it, and let it grow. So throughout the course of the day, we're collecting the weight every minute in the lysimeters and taking 15-minute averages. And that basically we're getting how water use changes over the course of the day inside these lysimeters. Of course, if you irrigate, you can see that. And if it rains, you can see that. And then we have a drainage system on the bottom of the tanks too. So whenever the water holding capacity is exceeded inside the lysimeters, it goes into the drain, and we have an automatic pump that then pumps that water out and calculates how much of that water is being removed from the lysimeters from drainage. So here's an example of the evapotranspiration that we've gathered during the first year of study. So this was the first year after planting, 2020. So the white dots is I have ETO here, but this is actually ETR from the weather station in Aurora. And in the black dots is the evapotranspiration of the blackberry plants growing in the lysimeters. So what you can see is that ETR in this case is higher than ETC, so it's going to be less than 1. But you can also see that as ETR goes up or down, the ETC is going to mirror that, and that's exactly what we'd expect because that's based on what's happening with the weather conditions. So what we do then is we then take the ratio of ETC to ETR to calculate KC. So then we can basically get a crop coefficient from every day during the growth of the plants. Now we know from other lysimeter studies that crop coefficients are well correlated to canopy cover. That's primarily what's driving water use in plants. So we also want to make sure we know how much canopy cover we have throughout the season. And the way we're doing that is using our drone. So here you see an image, an NDVI image that we use with our multispectral camera that's attached to the drone so that we can then carefully monitor how much canopy we have as the plant grows throughout the season. So here on the top left is an example of a crop coefficient curve from the first year. Now we wouldn't expect that nice curve like like they use in Agromet, where it goes up, levels off, and then sort of slopes down at the end during the first year because the plants are young and they're gradually developing their canopy. So that crop coefficient is going to continually increase throughout the season. So you can see that's exactly what happened here, with the exception that when we tied the primocanes up to the trellis wire, you kind of compress that canopy. You don't have as much cover at that point, so the water use goes down, but then it starts gradually building back up again. So this past year, the idea with lysimeters is you want the plants to look exactly the same inside the lysimeters as they do outside the lysimeters. So here's a photo in the center that was taken from the harvester, and you can see that basically where that white box is, that's a lysimeter. And you can see the plants, you can't tell where the lysimeter is versus those outside of the lysimeter. And that's ideally what you want, because if you have plants that are bigger in the lysimeter than outside the lysimeter, you get what's called an oasis effect. And your plants will actually use more water than they would naturally if they were, all the same size. And of course, if they were smaller, they'd use less water. But we did have that heat dome event, and you can see on the top right there, that was taken on July 7th. There's a picture of Jess at the Caneberry Field Day, and you can see the scorched leaves at that time. And we did lose some berries at that time. We lost about 30% of the fruit during that heat event. So some of the questions that we're now able to answer with this lysimeter is, okay, so how much water are they using not only when they're young plants, but we're going to continue to follow it, see what happens as they mature, so it can develop crop coefficients from planting to basically to full maturity. We're also able to answer questions such as, during this heat event, one thing that's often said is that berry plants will shut down when it's hot. Well, what we were able to see from the data from the lysimeters is that they don't actually shut down. I mean, they're pumping away, they're getting water out. They just can't keep up with the demand, so consequently they become stressed. So they don't actually close down their stomates during a heat event. They just can't keep up with it. As we continue on with this project, we do have the 2 lysimeters. Our data was very similar between the 2 lysimeters because the plants were more or less the same size. So there's not a lot of difference between the 2. So what we'll start doing the next year is we'll start comparing the traditional training practice, which is basically training cutting off the old floricanes and training up the new primocanes in August versus new over old, which is what Amanda and Bernadine were looking at, and looking at what effect that's going to have on water use, where basically you take the new primocanes and you just wrap them up over the old primocanes. And some growers are doing this. And after we do that, the following year, we can start looking at developing a water stress coefficient for blackberries too. We can well water one lysimeter and stress the other. Now, of course, it'd be ideal to have a lot of these lysimeters out there, but logistically, that would be very difficult to do. Financially, it'd be difficult, but logistically, even more difficult. With the drone, we also have a thermal imaging camera on it. And what thermal imaging does is it's basically measuring the temperature of the leaves. So when a plant is stressed, doesn't have enough water, it closes its stomates to try to reduce water loss. And when that happens, the heat goes up. So with the thermal imaging camera, you can actually fly over the field and look for stressed plants. What we're doing is we're developing a crop water stress index for this thermal imaging that we can then use the drone to actually see what the water status of the plant is. We'll be able to relate what the temperature is on a given day based on the weather conditions and relate that to the water potentials so that, conceivably we could fly over a field and actually see what the water potential is in different parts of the field. Now for growers, that'll be great. If we have a consultant that has a drone and is capable of doing this, they could fly over a field and it can actually see, parts of the field that aren't getting enough irrigation or whether you're irrigating properly and so forth. Now, of course, having these crop coefficients is very useful too for accurately estimating your water requirements in the field and preventing water stress and maximizing your growth and production in the field. We will tie all this information. We'll feed this into AgriMet, AgWeatherNet, as well as the mobile app irrigation scheduler that was developed by Troy Peters, so that this information is easily available to growers. So basically, we'll update those theoretical curves with more realistic curves. And we're doing the same type of work now in blueberries, where we'll have, in this case, we're gonna put in at least 6 lysimeters, 9 if I can get more money for it. So we'll have an early, mid, and a late season cultivar, lysimeters for early, mid, and late season cultivars of blueberries, and which I think is particularly important in blueberries because their water use in an early season cultivar is very different than in a late season cultivar. Now, in terms of other treatments that we're going to be starting posing now that the plants are mature, we're going to start to look at post-harvest irrigation and whether that's a negative or a positive effect. But growers are all over the board in terms of what they do. Some growers, they'll irrigate the whole season. Some growers will irrigate up to fruit harvest. Some will irrigate up to and through fruit harvest and then stop. And then some will actually not irrigate at all. I think there was a survey out, and I think I don't want to say, it's about 10% of the growers don't even irrigate their blackberry fields here in Oregon. Well, we had done a study in organic blackberries a few years ago where we had treatments where we either fully irrigated after harvest or we did not irrigate after harvest. And some of the benefits we saw was reduced weed growth, of course, because you weren't adding water after harvest. It had little to no effect on fruit production by shutting off irrigation after harvest, but it also reduced winter injury. And that was something that growers had said that, if we stop irrigating after harvest, it hardens the plants off and they do better in the winter. Well, in this case, we did show that that did happen. And of course, not irrigating, you're going to save water. In that particular study, we estimated that we saved about 67,000 gallons per acre. So we're going to repeat that work. In that case, the field was sort of on a lower part of a hill, and I wondered if water was, sort of running down the hill and feeding those deficit treatments. In this case, we're on top of the hill in a more sandy soil. So we're going to have 3 treatments where we fully irrigate, we do reduced irrigation throughout the season, or we do no irrigation after harvest. So basically from August, September, we're not going to irrigate, and look at what happens in those plants. So in a nutshell, this is basically what we've done and where we're going from here. So any questions? Yeah.
On the dry farm plants, were you seeing big yield swings compared to irrigated plants?
So people that are doing no irrigation, talking about, do we see yield swings? Yeah, it's going to very much depend on the year. And people who didn't irrigate, obviously, this past year suffered almost 100% crop loss in some cases. Yeah, because that can be a problem. I mean, I don't know. I hope this heat event we had in June will never happen again. I hope it's an anomaly, but who knows? Yeah, Randy.
How low have you guys been able to get the cost of your homemade lysimeter?
How low? Well, when we built these guys, I think we paid about $10,000 just for the construction of the steel. So the inner tank and the outer tank was $10,000, and then you add another about $5,000 on for like the all the electronics, the data loggers, and so forth. So about $15,000, and then of course there's all the wiring and so forth. We had to also pay electrician to send the electrical power out to the lysimeters. Now, of course, we just built some more for the blueberry lysimeters. They're smaller, and the price went up to, I think it was $13,000 in each for those because of the increase in price of the steel. So compared to $50,000 in 2000, we got the price down to about $10,000. Yeah.
So what's the square footage or the diameter of the root zone that you are allowing the plant to grow in?
Yeah. So
is that in any way limiting?
That's ideally you don't want to limit the root zone, right? So we know that at this site that the roots go about 3 feet deep in blackberries. So our lysimeter tanks were 3 feet wide, 5 feet long, which was basically the normal plant spacing for blackberry, and then 5.5 feet deep. So we feel that the roots they're not restricted in these lysimeter tanks. I mean, they're not as big as the ones we had in California, but they're not restricted. Blueberries, we're not going to need to go as deep with those. So in that case, our tanks are only 3.5 foot deep, but again, they're 3 by 5. Even though the plant spacing is 3 feet, we kind of have 5 feet to be able to get the raised bed inside the lysimeters. Yeah. So you had a question?
So the automated irrigation system, would that be when you put the human entry of the ET, or are you saying that the program will calculate your ET for you?
So for calculation of ET, it's all automated, and it's based on weather conditions. Yeah. Is that what you're talking about and like an agriment?
No, you mentioned the automated, using this for the automated irrigation.
Oh, no, I don't think I said that using this to automate irrigation. It's just what we do is have that information available for growers to be able to see on this day, this is how much water I think my plants used. Of course, it's up to the growers to kind of adjust up and down depending on how their field's doing. Yeah. No, we're not automating. In this case, we are not automating. Not that it can't be automated, and we do in our experimental fields automate all the time, but I don't know how if I was a grower, I don't know if I would trust that much to automate it. Julie.
Dave, on trailing blackberry, do you run a single drip line or a double drip line?
On trailing blackberries, what they typically do is just one single line hanging from the trellis wire. Same thing with in a lot of cases in raspberries, they'll do the same thing. Unlike blueberries, where they'll have dual lines, for various reasons. Not that you need that many lines for the irrigation. see it.
Are they, like single emitters on those plants or?
Yes, so basically they're inline drip emitters, so it's drip tubing, and the emitters, I think, I don't remember what these were spaced at. Can't remember if it was 24 or 30 inches or something like that, yeah. Don't think I have any, if anybody had typed in a question, it's online, it's not showing up, so I apologize for that. All right, well, thank you, guys.
Okay, it's 4:30, so I guess that we can start and proceed with this talk. Good afternoon or better, good evening. Gabriella Tait, postdoc here at the Oregon State University in Von Walton Lab. And today I'll briefly describe and trying to summarize the data collected this year on a new deterrent we created in our lab and its efficacies against Drosophila suzuki. So as you know, Drosophila suzuki is an invasive species coming from the Southeast Asia, which arrived here in the United States as well in Europe 10 years ago, well actually 12 years ago. And the problem is due by the fact that females are able to insert their ovipositor inside berries, ripe berries, and for this reason we had an economical impact on our industries. Our growers currently base their control of Drosophila suzuki on pesticide. So the goal of our research as scientists is to try to find new solutions in order to reduce the impact of toxic pesticide and give the access to our growers to new approaches. So just to briefly introduce you to the concept of a deterrent. A deterrent is different with a repellent. If with a repellent you totally keep the insect away from the berries, so there is no contact at all between the berries and the insect. With the deterrent, you still have a contact between the substrate and the insect, but the time spent on the substrate is less. So we should have at least in the perspective, a lower number of eggs released on berries. So this is our deterrent. And the objectives of our research were two, at least for this last summer. Test our formulation at semi-open field conditions, and then try to identify by GC the volatiles emitted by the deterrent. So this deterrent is the result of 4 years of work. Myself and Professor Von Walton, we are the inventors of this new technology. Coming with this new technology meant a lot of work, a lot of effort, a lot of research has been done. I just can tell you that we tested more than 50 different compounds. So we ran almost 100 at least lab tests. For each ingredient I tested a different concentration. And yeah, it's more than 3 years of work, not just in the lab, but also in the field. And luckily, over the last 3 years, we have been able to improve the quality of our deterrent. So let me briefly introduce you to our trials. So we were able to test our deterrent in a semi-open field conditions. We have some 2 acres actually of blueberries crop here at Lewis Brown Farm, a few miles out of Corvallis, and thanks to which we are able to test our products. So we wanted to build big cages and in a way that we could treat some bushes under the cages, release flies inside, and twice per week over a period of 1 month in August and 2 weeks in September, collect berries and detect the number of eggs released. So in August, the idea was to just treat 1 bush out of 5 in order to see if the efficacy of the deterrent was if the deterrent, its volatiles were able to cover and protect also the other 4 bushes. 100 flies had been released per cage, 50 females, 50 males, 2 sampling per week, 250 berries per cage. So it's been quite a lot of berries to count and check, and 3 kgs per treatment. The concentration of our deterrent was 0.075 volume to volume. So we're really excited about, because when we started 4 years ago testing our formulation, we were talking about 6% volume to volume. Then after a lot of work in the lab, but also out in the field, we were able to improve the formulation, reduce the quantity of powder, and therefore we were able to identify the 0.075% as the best solution. So in this case, here we are just talking about powder. Is a mixture of different powders and water, nothing more. In September, we wanted to test the efficacy of the deterrent, as is it, by adding as well an adjuvant in order to see if it was possible to better cover the berries with our deterrent. And also the second parameter we changed this time was to treat all the bushes under the cages and not just one. And but we were able to release the same number of flies and still collect 250 berries per cage. As you can see here, we tested 3 different concentration from 0.125 to 0.5. So the real concentration of the deterrent was still 0.075 plus. We added a different concentration also of adjuvants. Here our result. So in 4 collection out of 6, we were able to detect lower infestation in terms of eggs in the treated plants and not in the control. While during August 13 and August 27, we actually recorded a higher number of eggs in the treated bushes and not in the control. So we still have to understand what's it happen. Multiple parameters could have affected the efficacy of our deterrent. But overall, we see that the level of infestation in the treated berries were definitely lower. In September, we were able to collect berries only 3 times after the application of the deterrent, because it was the end of the season, and we were really we were lucky to at least try to perform the experiment. But still, 4 days after the treatment on September 9, actually we detected more eggs in the treated bushes and not in the control, while on September 13 and 17 we recorded a lower number of eggs in the treated bushes. To the best of my understanding, this happens because the volatiles and the deterrent need time before starting to be effective against Rhodophylla suzuki. So what I'm learning from our experiment is that when you spray your deterrent, you need to wait a little bit before starting seeing some results. So, and the second goal of our project was to identify key volatiles emitted by the deterrent. Is a work in progress. I'm working on it. So what we can see here in this picture, we were able to identify at least see some pigs originated from both females and males. We still have to do the identification and characterization of these volatiles. What do I mean? I mean that we see in this picture that there are responses, but we don't know the name of the compounds. It's something I have to do with a technique named mass spectrometry, gas chromatography, which will allow us to give names to these peaks. The final goal is to work then to identify these volatiles, put together the volatiles at the right concentration, and instead of having a deterrent to spray on berries as a full coverage technique, we would like to create some sort of dispenser to place, I don't know, every 10, 50 or 20 bushes. We still have to verify in order to a dispenser that can release the volatiles without having not impact on the berries as the deterrent could have. Who knows? So these are results not collected by our lab, but from our colleagues in New Jersey. As you can see here in this picture, OSU, which is actually our deterrent, performed really well compared to other treatment and to the control. So it was really exciting to get great results from others that is not us. And also, I just wanted to say that all the results we collected, all the berries we checked in our lab um, were not checked by myself or my professor von Walton, but always by student and another postdoc supervised by another postdoc, Sarah Mermer, in order to make sure that our feelings were not involved because we are the inventors. So I just wanted to specify this. But I guess that these results help to understand that this deterrent is really something real, not just in our imagination. And which are the possible benefits for our industry? I guess that it's possible, it's real. The idea to have this deterrent out in the future. We have growers willing here in Oregon as well in California and other states to test under open field conditions this deterrent. The point is that right now our industry is based there control on just few pesticides like Venerate or Grandivo, Jetag, and Trust, and we know that a resistant genetic process is developing, is happening, and for this reason we need to diversify the quantity and the quality of product we spray on our berries. So currently, after 3 years of field work, we didn't identify any phytotoxicity on the berries. The taste, the flavor are still the same, at least in our mind and in our mouth. But we need to involve more people. We need to create like a panel taste in order to have external people have an evaluation on the taste of the berries. We can say that we have a comparable efficacy of this deterrent compared to available marketing pesticide. The longevity is still between 4, 5, 6 days according to the weather conditions. For sure, would include a similar application cost. But as we were saying, we still have a lot of work to do. We want to identify a key volatile. We would like to have a final product that is not something that you spray on berries, but is something that it's emitted by an external object that does not impact berries and workers out in the field. So for the next year, open field trials are necessary, and as I was saying, we have people willing to test it, and yeah, this is it. I guess that if there are questions, we can go ahead. And thank you for giving me the possibility to present our data. Thank you for funding our projects. And yeah, this is it. Thank you. Thank you. Yeah, I guess that we can look at the chat if there is something. Hopefully not, so we can go home and start our evening. I mean, no, for sure, if there are questions, I'm more than happy to at least trying to give an answer, because sometimes, we don't have answers, right? We are scientists and we need sometimes to say, I don't know, but thank you for the question. But, yeah, definitely. So, yeah, it seems that people, yeah, I guess that the three of us. So if you guys don't have any questions, I let you go. Thank you. Thank you. Thank you so much. Thank you. And thank you, John, for the assistance. Bye-bye. Okay.
Okay. Yeah.
Hello, everyone. My name is Kim Hummer. I'm the research leader and curator at the National Clonal Germplasm Repository in Corvallis, Oregon. Today, I'd like to present to you a study that I've most recently completed concerning diversity in the cultivated strawberry Fragaria cross ananassa. We did a big study looking at phenotypic characterization of a strawberry diversity collection. This collection represents part of our strawberry cultivars at the National Clonal Germplasm Repository. I'd like to recognize my colleagues and co-authors who assisted greatly in this project. Dr. Nahala Basil, who is our molecular geneticist, Dr. Jason Zern, who was a postdoc with us, but now he's at Kansas State University. Beatrice Amiot is with the Agriculture and Agri-Food Canada in Nova Scotia. To describe the diversity of the strawberries that we selected for this study, I have several different tables here. Strawberries that we were looking at were from 17 different countries and 19 different states. And as you can see by the totals there, the majority of the cultivars we observed for the study were from the United States. And the main breeding programs that they came from, University of California at Davis, we had 65 accessions. The USDA breeding program in Maryland, there were 33 accessions from there. And in Oregon, we had 21 accessions from the USDA program there. This first column on the left describes what we just talked about, but in a graphical sense, you can really see the 175 cultivars of the United States, much more than others, although there were representation from many other breeding programs around the world. The second column shows that the majority of the cultivars that are in this particular study were released between 1991 and 2000. So the cultivars that are examined are primarily either first or second generation from the latest patented materials that are now in commercial supply. The third column shows that mostly we looked at named cultivars, although there were breeding materials and a few wild materials collected also. Our whole staff came out to help with the planting of the strawberries. If you have 12 rows, 75 plants in a row, and 3 replicates, it's quite a big undertaking. There were 3 blocks in the planting, and I gave them colors. The blue block was the first block. It's the first 4 rows. The red block was divided, the 2 rows on the west side of the irrigation row, and then 2 rows on the east side of the irrigation row, and the yellow block was further east. So there were 3 blocks for the study, and it was a randomized complete block design. And we collected 15 phenotypic and phenological descriptors on these strawberry cultivars. Fruit samples were collected and separated into groups for analysis. Samples were first measured, weighed, and photographed. Fresh fruit were photographed at the laboratories at the NCGR. Then samples were divided to be frozen for sugar and acid analysis, and a second group for anthocyanin analysis that was to be done at the Linus Pauling Institute at Oregon State. The remainder of the fruit was put aside and kept for seed extraction. Here's a huge table that shows all 15 types of information that we were collecting about the different cultivars there. Here, I'm just going to speak briefly about the weight and also the largest fruit that we observed was 45.1 grams. And the largest total harvest was a little over a kilogram, 1,029 grams. I'm going to jump over here to the runnering information. This was one thing that other people have just done, high, medium, or low runners, and we wanted to make it a metric that was more numerical in estimation. And so what we did was we collected all the runners from each of the plants during the week of July 7th. So it was after they were done ripening. For the June ripening period, we collected the runners the week of July 7th. We put the runners end to end, and the strawberry that had the maximum amount of runners, there was 112 meters of runners, and there were 492 runners on that plant. So next, we'll look at fruit quality traits. And I just made this a very brief, rough and tumble presentation, so I'm not going to go into great details. But this set here that we have on the left, these cultivars had some of the lower qualities of fruit traits, while the ones circled on the right had the best fruit quality traits. In another grouping, we looked at the shortest ripening time. These cultivars had the shortest ripening time, and the ones on the right were longer. Longer time was needed for their ripening. So with these 15 traits, we performed a Pearson's correlation on the data. And I present just 3 examples here. The first example is at the top, where we were comparing harvest date by bloom date. And you can see that that's slightly positive. You can see it by the tan color in the diagram and the upward trend if you were to draw a line on that cluster map there. The second set of data, days to ripening, we looked at days to ripening as compared to bloom date, and that was a negatively correlated trait. You can see it trending down. And here's another one, days to ripen and harvest date was positively correlated. That's on the bottom. And we did this with each of the 15 traits, comparing their correlations. There was a trend in over the breeding decades that we looked at to move from globose to conic and conic wedges shaped in the strawberries. The berry weight on the bottom graph there was not clearly seen to decrease significantly over these decades, given this particular set of cultivars and the studied climate conditions. I want to point out some quality trait stars. The 3 cultivars with the best quality traits were Independence, Oris 2427-1, and NW 90054-37. It was interesting that Independence had the best of both worlds with good fruit quality traits. And then if you look at that histogram there on the bottom, it also had an abundance of runners by the 7th of July. By the way, Urus 2427-1 is a full sib of Mary's Peak, another cultivar that was released by Chad Finn. Yes, Mary's Peak was in the test also. Other quality stars, the ripening speed, these fast ripening strawberries included Oso Grande, Cuesta, and Pinnacle. These three cultivars had a quick start and were the ripening stars for the study. But if you take a look, they put their carbohydrates into the fruit and did not produce as many runners by the July 7th date when we collected them. Now I want to take you over to Grin Global. One thing we did was upload all of our information on each of these 284 cultivars to Grin. And some of you may not have had the opportunity to go up and search Grin. So I thought I would give you just a brief introduction as to a start of looking for information on our information there. So, because we have lots uploaded there. So in this case, let's go, sorry, wrong key. We have a descriptor tab. If you go to the main Grin search page, web page, this page is the main search page. And at the top, there are tabs. So if you click on this descriptor section, you go next to an area where there's an opportunity to select descriptors for different crops. And in this case, our crop is strawberry. So if we type in strawberry in the search box, what comes up is the strawberry descriptors, and that's highlighted there. And there's a number of strawberry descriptors that scroll down to the bottom, but I just have a fixed slide. So just for an example, you can see that there's total anthocyanins in milligrams per 100 mils as the value. So if we were to check that, you see there's a little box that you can check there. And if we search for that descriptor, you get to the study group information. And you can see that we put information here for the study of 2019 and the one of 2020. And in 2020, there were 272 accessions with information on our total anthocyanins. So if I were to click that blue number that says 273 accessions, then it would take me to the accessions on the website. And as another bonus during this study, we took images of the fruit and we've uploaded all those images there on the website. And we've also uploaded images of flowers, images of plants, and that kind of thing. So once you get to this page, there's a blue arrow there that I'm using to indicate the value. If you were to click on that value tab, it sorts the values according to the highest value. So this particular screen shows the highest value of the total anthocyanin content. And you can see some of those with the high quality traits that we were just talking about are there. Now, if you click that value button once again, it sorts it in the opposite direction. So you can see that the white fruited strawberries have the least anthocyanin content. They're at the bottom of the scale, and white ananas and white D happen to be strawberries that are low in anthocyanins, and they're at the bottom of the heap. Now, I do want you to see the blue shopping cart there, because where there's a blue shopping cart, you can take your cursor over and click on that shopping cart and put an, if you want to make a request for Weiss ananas, you can click on that shopping cart. It goes to your shopping cart. You can go in and make an order for the strawberry runners. In our case, we distribute strawberry runners at the end of the summer because we cut after the winter, we cut the plants back for winter, and then they grow new runners, and that's our distribution product that we ship out. So that's just a little, oh, here's one more little interesting thing. If you have multiple traits that you want to search for, you can search for multiple traits. Remember back at that where there were little boxes, you could check off the box. So say we check off the largest berry weight box and the total anthocyanin box, and then it gives you an option to choose values. So say you want greater than or equal to 20 grams size of fruit, and say you want greater or equal to 20 milligrams per 100 grams of fresh weight, then it will come up with a list of strawberries that meet that criteria. So in this case, there were like 52 accessions that fell into that category, but it gives you them in sets of 10 because that's all it fits on a screen. And I've just taken a screenshot here so you don't see all 10. But it you can choose accessions from our collection that meet the criteria that you're looking for. And if you choose others that are resistant to botrytis, or maybe, so maybe there's diseases or other qualities that you want to select for as well. So it's a very useful tool now that we have not only the accessions, but the characters of the accessions are being uploaded to GRIN. And this particular study was an extremely useful one to look at the 280 that we looked at. So in conclusion, one of the accomplishments was uploading the traits for 284 cultivars to our GRIN global database. Again, for us, we had extra seeds for cultivars. Open pollinated seeds were sent to Fort Collins as a backup germplasm. And in fact, these so much seed was sent there, some of them can be forwarded to Svalbard Seed Vault in Norway. And so that's a second backup option that we have. And then, of course, there were the data that we obtained. Largest average runner production, 112 meters, which is a lot of runners if you think about it. Largest harvest per plant was the kilogram of fruit per one plant. And this is just the June bearing part. We didn't have enough resources to also look at the ever bearing. And strawberries that were consistently above average fruit quality characteristics included the three I mentioned: Independence, Oris 2427-1, NW 90054-37. And then strawberries with the fastest, most consistent harvest under our conditions were Oso Grande, Cuesta, and Pinnacle. So I would like to thank all my wonderful folks that helped with the technical aspect. In 2018, when we started this project, Dr. Lin Chang was a visiting scholar with us for 4 months, coming from Beijing, their gene bank in Beijing. And also, I'd like to thank Laura Duncan, Brandon Harms, Gabriel Flores, Ryan King, and Todd Anderson for additional help with data collection. Bob Durst was overseeing the Linus Pauling anthocyanin study that was done, and Zach Weigerd helped with fruit chemistry in the OSU Food Science and Technology Department. Last but not least, I'd like to acknowledge the funding sources from the Jewish Communal Fund, USDA ARS Small Fruit Crop Evaluation Grant of 2019, and the Northwest Center for Small Fruits Grant of 2020, and our USDA CRIS as well that supported this research. So thanks very much, and I'm open to questions that you may have about the study. Sure, go ahead. The question is, what about the fruit shape, and why did people develop strawberries that have a changed fruit shape over time? I think it's because the wedge, the conic wedge, you can get a much larger fruit. And that seems to be where the industry is going, where the market has requested larger, redder fruits. And that shape seems to be pleasing to the consumer.
What about, you measured a lot of fruit quality characteristics, but what was the best tasting?
This particular study, I did not sample, or we did not have a person or group or that sampled these. Yes, it was. The last time I was involved in a study in 1985 where we did a set, well, 250 at that point in time. And that's where I developed my favorite strawberry was Holiday. And that one I did taste all of those guys. But also, you know, there's taste panels and things like that. And we didn't, we tried to make sure there was sufficient fruit to go to the fruit analysis part. And so we didn't really eat the germplasm, although a few things were. Now, I will say we had a large number of Marshall, the true Marshall plantlets, and we used them in the guard rows. And so I made sure that all of our staff got to take home fruit of Marshall strawberry as a benefit of helping out with the strawberry collection. They were very tasty. And the challenge with Marshall is that it is not a firm fruit, and you just have to enjoy it right there in the field because it's dripping down your chin kind of taste. So we didn't really explore the flavor aspect of it.
Not so much a critique. I think about it often when you have the older lines that are probably selected mostly on flavor. And now we try to boil it down into a few numbers. I had another question about, you showed some interesting correlation, trade-offs, or I assume a lot of this phenotyping is going to go into the mapping, the genetic mapping.
Yes, our goal was to, because we have a corresponding genetics project that's going on right at this time too, and they looked at these same, actually not only these, but all of the cultivars in our collection. So we are working with Nahala Basil and her cadre of geneticists to do essentially a GWAS type study looking at the phenotypes and the genotypes. And this is just the phenotypic start of it. So we hope to link the genetic, the genotypes with particular traits. One of the desires, and when it was originally planned, were to look at more disease traits. And we had planned that some other folks would be involved in the study that could do that pathology. And so they were going to look at the easy, I shouldn't say that, but it's a little easier to, if you're looking at genes, to look at disease resistance and not, since the gene for gene theory and all that. But unfortunately, that fell apart. So we did the phenotypic study in our own backyard, and at least we came up with data that will be interesting, but a different set. So another day we'll have to do some disease markers and other things to combine into that.
With that longer term project, I guess in looking at those correlations, do you have any sense of if they'll be able to answer how much of those correlations are genetic or just differences between breeding cohorts and different bottlenecks in those?
Well, it's not just a matter of the gene sequences. It's interesting, in the strawberries, we have 400 years of data, or at least word of mouth of what the pedigrees are. And so we're working with the pedigrees and parentage analysis and software for that, as well as looking at the sequencing data. And so where things line up, and we have, as you saw, there's some very old cultivars in our collection that was in the study, and progressively every decade too. So we can go back in the parentage and take a look, and also with the sequencing, is it matching up? So there's a lot of different ways to look at. Another neat thing that we're working with is the percentage of similarity of the DNA, and that's bringing out some really exciting findings as well. So this, we're hoping for a couple papers to be coming out soon regarding this.
So we can order plants?
Yes, you can order plants. Right now, we cannot supply the plants until next August, because after the runners grow, but you can look through at your leisure at the different quality traits and the images that we got. And so we're trying to make GRIN as, and not only for strawberries, we're trying to do this for the blueberries, raspberries, all the other berry crops, also for our pears and hazelnuts. And so the goal is to make GRIN the go-to location for information for plants in the germplasm system. So we do have USDA patented materials, because the USDA breeders deposit in our collection. Breeders that choose to protect their material through a plant patent, we don't get till it comes off a plant patent. So the reason we have older UC cultivars in our collection, as soon as they come off patent, the Plant Materials Center down there is like, hey, Kim, can you take this for the repository? Gets it off their hands. We maintain it. And but they don't want it a day sooner, because early on in the 90s, some other people had generously donated UC cultivars to our system. And I got a letter from the Regents of California lawyers And that took up the whole first page of all the lawyers. And it was like cease and desist from keeping patented University of California strawberries because they're protected. We don't want you to have them. And so we took that all out. Now, as soon as it's 20 years later, and they're like, oh, please take these formerly patented cultivars. So we do not have the latest patented information because we don't have the plants. They have to be in our collection, and then we can add it to the GRIN system. We are not a Clean Plant Network program per se. The Clean Plant Network have a small amount of cultivars, maybe 10 or 20. We have 500. So we're also right in the midst of getting a rebuild on our greenhouses and all because our roofs have blown off in the winds and whatnot. They're 40 years old and we've got to replace our facilities. Once we get an integral system again, and we do have pathological knowledge base, we know how to do ELISA tests and PCR tests, and so we'll get back into a routine system of testing. And when you talk about clean, once something tests clean, there's a timeline, and you have like 3 years until you have to retest it, maximum of 3. And the Clean Plant Network tests them every year. Well, we don't have enough resources to test every plant every year, but we endeavor to send as clean material as we can to our requesters. Well, Mary Peterson. No, she didn't ask a question. She thanks me for the presentation. Thank you, Mary. Okay. Well, thanks everyone for hanging around and listening. And it's been an interesting Northwest Center meeting, one for the record books, I think. Yes, they're available in the public domain. Yeah. Yeah.